My pleasure in welcoming you all. The This webinar has been supported by the Sustainable Vaccine Manufacturing Program that we have at PATH, that's jointly created by the Bill and Melinda Gates Foundation and PATH to provide technical assistance to DCVM and manufacturers and help to sustainably produce safe and effective vaccines of international quality. This webinar has been organized with the objective of supporting vaccine manufacturers. And the scope of this webinar is to provide manufacturers the broader understanding of aluminum adjuvants from a perspective of chemistry, immunobiology, manufacturing, characterization, formulation, and we will also talk about challenges that many manufacturers face. So why do we need adjuvants? We need adjuvants for multiple reasons: to enhance the immune response for weak antigens. We may need to increase the duration of the immune response or reduce the amount of antigen needed in the vaccine. We also want to influence the magnitude and avidity where different mechanisms that different adjuvants can use. Enhance the immune response in elderly or in infants who are immunocompromised. Then you also can direct the immune response, either an antibody response or a cell mediated, to match the need of the protection that you need for the particular type of vaccine and prolong the duration. There are multiple licensed vaccines. Most common of all is the aluminum salts that we all know of and we're going to talk about. There are others too, such as MF59, ASO3, ASO4, CPG, and which we won't go into. But it is important to understand there are other adjuvants that have been licensed more in the recent years. I'm unable to move the slides, Laura. The Here's just a slide of the aluminum content in vaccines, and this is just to give an overview. The amount of aluminum in vaccines can range from almost 0.25 milligram per ml all the way to 1.2, 1.5. So basically, this is to let you know that the amount of vaccine and the aluminum in vaccines can vary, and not only that, the allowable limits are dependent upon regulatory agencies. Um, Going to the next slide. Can we have the next slide, please? Yes. So today's agenda is we have just provided you a little introduction. We have several talks by renowned speakers who are very much in the field and will provide you a lot more of information and learnings. We have a talk on chemistry and immunobiology of aluminum adjuvants by Harm Hogenish, who is a professor of immunology at Purdue University, followed by a manufacturing by the Croda. And we all use the aluminum salts from Croda, Lauren Duron, let's talk about. And next is going to be Dr. Rajesh Gupta, who's going to talk about aluminum adjuvants and what he's learned from it from the multiple years. We'll have a short break, followed by the next slide, please. Yeah, by case study. No, can you go back to the previous slide? The next slide, please. I, Previous slide, please. That, 2 slides and yes. The next slide, the next talk is going to be on case studies on aluminum adjuvants by Dr. Robert Citron, who is also from PATH, and followed by formulation strategies to enhance the immune response to quality to aluminum-containing adjuvants by Dr. Christopher Fox, who's from the Access to Advanced Health Institute. And the last talk will be Building on a Solid Base by Dr. Derek O'Hagan, who is a senior advisor from GSK. What we'd like to do is that we'll keep the questions in the chat box so that during the talk, it may be difficult to respond. So we'll keep it to the end. If there is speaker time, we'll do it after each talk. Otherwise, we will wait till the end of the session. We have a break of 5 minutes in between. So before the break, we can take some questions, or at the end, we have time for questions. But if the speaker is able to address questions in chat, we will do so. And we would encourage everybody to mute their phones. Can we go into the next slide, please? Few house rules that we have is the meeting is going to be recorded and will be made available. So if there's anybody who is interested and was not able to attend, please let us know and we're happy to share it. Then please be free to use the chat feature to introduce yourselves, to have questions and anything that you would like to share. And then we'll definitely, of course, have to have silence on phones and refrain from checking emails so that we can really focus. And mute your phone except when you're speaking. And Laura McCormick will be the point of contact for any other questions. So without any further delay, let me introduce you to the first speaker, Harm Hoganish. Yes, would you please take over?
Yes, good morning or good afternoon. I would like to thank Lakshmi for the invitation to speak here for PATH, for the organization of this workshop, and Laura for the technical support and making this all possible. So my name is Harm Hogenes. I'm a professor at Purdue University in the Department of Comparative Metabiology. And I will talk with you about some aspects of the chemistry and immunobiology of aluminum adjuvants. And if I can. Yeah. I've done some consulting for a chemical company in the past year called Aurorium. It's headquartered in Indianapolis. It's a large company that acquired a smaller company that makes aluminum adjuvants, previously known as Revice, but falls now under Aurorium. So what I'll do today is I'll talk about, give you a brief introduction about aluminum adjuvants, and then we'll talk about the chemistry of aluminum adjuvants, or absorption mechanisms, how absorption can affect the stability of antigens and how it can affect the immune response, and then also importantly how you can change the degree and strength of absorption to affect both the stability and the immune response. And then I'll finish up with talking about the mechanisms by which aluminum adjuvants enhance the immune response. So aluminum is actually the third most common element on Earth after oxygen and silicon, and it comes in a lot of different forms. I'll mention a couple of them here before we delve into the aluminum adjuvants. So you'll hear and you'll have seen probably people refer to aluminum adjuvants often as alum, and that's just because aluminum adjuvants is a mouthful. And so people often refer to it as alum, but you should be aware that alum is really chemically a solution of aluminum sulfate, and often in the form of potassium aluminum sulfate. It is used to prepare aluminum adjuvants or to precipitate antigens with an aluminum adjuvant. So either aluminum adjuvants are synthesized and then they are mixed with antigens, or they are used to precipitate antigens. But the alum itself is not an adjuvant. It is used in a variety of applications. in addition to making aluminum adjuvants, including in the food industry, as this picture here indicates. Aluminum hydroxide, sometimes you see aluminum adjuvants referred to as aluminum hydroxide. Aluminum hydroxide is not an adjuvant. It's a mineral. The most common form is gibbsite. It's a highly crystalline material, as indicated in this EM picture. But again, it's not an adjuvant. And then in the literature, you'll see people refer to imject alum or use imject alum. That was a compound that used to be sold by Thermo Fisher or Pierce. I believe it's taken off the market now, but if you look at the somewhat older literature, you'll see references to imject alum. And this is an adjuvant that is used in preclinical studies, so in animal studies that lead to potentially human application. But it's important to recognize that this is not the same type of adjuvant, even though it has immunostimulatory effects. It's not the same adjuvant as used in human or, for that matter, veterinary licensed vaccines. In fact, imject alum it's chemically aluminum hydroxycarbonate combined with crystalline magnesium hydroxide, and as we'll see in a minute, is quite different from the aluminum adjuvants that are used in licensed vaccines. So I'll focus here on the aluminum adjuvant, aluminum hydroxide adjuvant that I'll refer to as AH, and aluminum phosphate adjuvant that I'll refer to as AP. So the aluminum hydroxide and aluminum phosphate adjuvants are really sort of nanoparticle-based structures. As you see here in these cryo-EM pictures here, on the left you have aluminum hydroxide, which is a fibril structure. On the right you have these plate-like particles, that is aluminum phosphate. And these nanoparticles, they aggregate, they form loose aggregates that are in the order of 1 to 10 microns in size. The aluminum hydroxide is actually chemically aluminum oxyhydroxide, and so again, it's not the same as the mineral aluminum hydroxide, AlOH3. Whereas aluminum phosphate is aluminum hydroxyphosphate, where the number of hydroxyl groups and phosphate groups can vary depending on the way it's manufactured, and so it's not a stoichiometric compound. The hydroxyls play an important role in aluminum adjuvants because they are metallic hydroxyls and can either accept or donate a proton, which means that the charge of the aluminum adjuvant is dependent on the pH. And so aluminum hydroxide adjuvant has a surface charge. I see here that the point of zero charge is obscured here on my screen, but the point of zero charge of aluminum hydroxide is around 11.4, which means that it's positively charged at the neutral pH, whereas the charge of the aluminum phosphate, the point of zero charge of aluminum phosphate is 4.5 to 6. Depending on the ratio of hydroxyls and phosphates in the aluminum phosphate, or the way it's manufactured. That's true for the commercial aluminum phosphates like Adjuvants, but you can actually create a wider range of the point of zero charge of the aluminum phosphate adjuvants. And then aluminum hydroxide is, as I already alluded to, it's a poorly crystalline material, as is shown here by the X-ray diffraction. It's sometimes referred to as pseudo-boehmite, whereas Adjuvants is amorphous. It doesn't have an X-ray diffraction spectrum. Another adjuvant that you may come across is amorphous aluminum hydroxyphosphate sulfate. This is an adjuvant that is present in many Merck vaccines. It is actually fairly similar to an aluminum phosphate adjuvant. It has the same morphology as shown here in these EM pictures. It has a plate-like structure. But it's different from the commercial aluminum phosphate like the Adjuvants because it has more hydroxyls and fewer phosphate groups, which leads to a higher point of zero charge, so about 7, as shown here in this graph and here. And that's, again, results from a lower number of phosphate groups, 0.3 versus the 1.1 ratio of phosphate to aluminum for the commercial Adjuvants or other aluminum phosphate adjuvants. And then finally, another form in which you may find aluminum adjuvants is by those that are created by precipitation of antigens. That's not a practice that is commonly used anymore, but that was the original way by which aluminum, the immunostimulatory effects of aluminum adjuvant was discovered. So in this case, you mix alum, so again, that's a solution of aluminum potassium sulfate with a solution of antigen. The antigen is often in PBS, and then you precipitate that with sodium hydroxide. And the result is that you get an aluminum hydroxyphosphate sulfate adjuvant that has similar properties to aluminum phosphate adjuvants. It's amorphous, but again, it's not a way that you often make adjuvant vaccines anymore because the composition and also the scaling of this product is difficult to control. Okay, so aluminum adjuvants have a very large adsorptive surface. It's calculated to be around 500 square meters per gram for aluminum hydroxide, and it's probably a very similar number, high number for aluminum phosphate. I'll show you some evidence here in the next slides that the adsorption is important for immunostimulation, but it depends on the degree of adsorption, the strength of adsorption. And then it also can affect the stability of the antigen. And again, I'll talk about that in more detail later. Whether or not the adsorption is important for the immunostimulatory effect, regardless, adsorption can be used as a quality control parameter of an vaccine. So you can measure the degree of adsorption of your antigens in the vaccine formulation. The WHO actually requires that vaccines for diphtheria and tetanus that at least 80% of the toxoids is adsorbed, and then the US has requirements of at least 75% adsorption of these toxoids. So how are these antigens adsorbed to the aluminum adjuvant? So the aluminum adjuvants basically form a solid surface to which proteins or glycoproteins will bind. The weaker forces are the van der Waals forces and hydrogen bonding. They have also hydrophobic forces can be involved. But the forces that probably are most relevant for in terms of vaccine manufacturing and the immunobiology are electrostatic forces and ligand exchange. Those are also the types of mechanisms that can be manipulated, and you can change the degree and the strength of absorption by manipulating the electrostatic charge and the ligand exchange, as I'll show. Oops. Oh, I see what happened. Okay. The slide doesn't seem to advance. Okay, here we go, sir. Okay. So electrostatic adsorption takes advantage of the fact that the aluminum adjuvants are highly charged. So aluminum hydroxide, as I mentioned, is positively charged at a neutral pH. So the yellow box Again, it's shifted a little bit here in the slides, but it's typically vaccines are typically formulated at a pH of 6 to 8, and at that pH, the aluminum hydroxide is positively charged, aluminum phosphate is negatively charged. Then next. If you then combine that with an antigen that has a low isoelectric point, so that it is negatively charged, that will absorb well to an aluminum hydroxide adjuvant. It will not absorb to aluminum phosphate adjuvant. And so we have done studies, for example, with ovalbumin as a model antigen, and you can see that ovalbumin it has an isoelectric point of about 4.6, so it's negatively charged at the neutral pH, and it will absorb well to aluminum hydroxide. It doesn't absorb to aluminum phosphate. Conversely, if you have an antigen that has a high isoelectric point, like we've used again a model antigen, a model protein, lysozyme, and that doesn't move forward for some reason. Let's see. Laura, can you advance the slide? Yes, thank you. The lysozyme is positively charged, and so it will absorb to aluminum phosphate, but not to aluminum hydroxide. It's repelled by aluminum hydroxide. And the next slide. The other important mechanism is ligand exchange, and that is a phenomenon that occurs because aluminum has a higher affinity for phosphate than for hydroxyls. And so if you have an antigen that has a terminal phosphate group, that phosphate will replace an hydroxyl, one or sometimes two hydroxyls for one phosphate. And that results in almost a covalent binding. This coordination of the phosphate with the aluminum is a very strong binding, and that is then the phenomenon of ligand exchange. I should mention that these same forces, electrostatic interactions and ligand exchange, also play a role when you make combination adjuvants. For example, if you combine aluminum adjuvants with monophosphoryl lipid A, MPLA, or with CPG, the electrostatic and ligand exchange forces also play a role in those kind of interactions with other molecules other than antigens. Next slide. So we've studied ligand exchange using, again, a model protein, in this case, alpha casein, which is a milk protein. 80% of milk proteins consist of casein, and it happens to have 8 phosphate groups. Some of them are in close proximity, as shown here. And you can incubate, you can mix the aluminum hydroxide with casein, and then that results in both electrostatic absorption because alpha casein is negatively charged, as well as ligand exchange absorption. And so in the next picture here, you can see that the interaction between casein and aluminum hydroxide is much stronger than when you use a dephosphorylated form of casein, where the absorption is just caused by electrostatic interaction. So the dephosphorylated form of casein, so without these phosphate groups, rapidly desorbs when you mix your vaccine with lymph fluid, so to mimic what happens at the site of injection after you inject the vaccine. You get rapid desorption of this dephosphorylated form of the casein. So the electrostatic bonds, even though you get complete absorption of the casein, the phosphorylated form of casein, it rapidly desorbs because the electrostatic charges interactions are not as strong as when you have ligand exchange. So with ligand exchange, you see almost no absorption over 24 hours of incubation. And so you can use this system of incubating your vaccine in either lymph fluid or something similar like serum or plasma, and then looking at the absorption of the protein as a way to address the absorptive strength, which is an important criterion for that has implications for the antigen stability, as we'll discuss in a minute. Next slide. So we can use this knowledge of the mechanisms by which the antigens absorb to alter the absorption, both the degree and strength of absorption, by focusing on the electrostatic aspects as well as the ligand exchange. And so I'll show some examples of that in the next slides. Next slide, please. So one way to change the electrostatic absorption is by treating an aluminum hydroxide adjuvant, or you can treat an aluminum phosphate adjuvant, but the effect is shown here for aluminum hydroxide. So aluminum hydroxide has a point of zero charge of around 11.4, and when you treat it with increasing concentrations of phosphate, you get replacement, increasing replacement of these of the hydroxyls by phosphate, because again, aluminum has a higher affinity for phosphate than for hydroxyls. And as a result, so you replace the surface hydroxyls by phosphate. As a result, you lower the point of zero charge. I should also point out here that you don't change the fibrillary structure of the aluminum hydroxide adjuvant, so it doesn't become an aluminum phosphate adjuvant. We refer to it as a phosphate-treated aluminum hydroxide. Next slide. Next picture. Yeah, thanks. And so when you do that, you can show that lowers the absorption by electrostatic absorption. So in this case, we used aluminum hydroxide and mixed it with albumin. If you do it in a buffer that does not contain phosphate, you get significant absorption here, up to 2.2 milligram per milligram aluminum. If you add phosphate, if you pre-treat your aluminum hydroxide with phosphate, then you lower the absorption. So the capacity of the absorptive capacity is decreased. In addition, the slope of this curve here at the lower amount of protein is an indication of the absorptive strength, and so you also decrease the absorptive strength by pre-treating the aluminum hydroxide with phosphate. And I know that Dr. Citron will talk more about the effect of phosphate on aluminum adjuvants in another talk later this morning. Next slide. And this electrostatic absorption has also been used to in a recent paper by Peter Kim's group at Stanford to specifically modulate antigens. So in this case, you don't change the adjuvant, but you change the antigens by genetically altering a protein. In this case, they used an Ebola glycoprotein, expressed it as in a trimeric form, and then they added aspartate residues to it by genetic engineering to the tail end of this glycoprotein, and then examined how that affects the interaction with aluminum hydroxide adjuvant, L-hydrogel. And you can see in the graph here that the glycoprotein itself doesn't absorb well to the aluminum hydroxide adjuvant, but if you increase the number of these aspartate groups, so you increase the negative charge of this protein, you increase the absorption, and they found that at 8 and 12 aspartate groups, they get the maximum absorption. But the question then, of course, is how does this affect the immune response? And so in the next slide, they did a study here where they immunized mice with either the Ebola glycoprotein or the glycoprotein with 12 aspartate groups. And showed that results in a tenfold increase of the antibody titers, about a log increase of the antibody titers, and a somewhat more modest effect on the neutralizing antibody titers. But clearly, increasing the absorption had a beneficial effect on the antibody response. Next slide. So. We've also looked at whether you can use ligand exchange as a way to affect absorption. And again, we went back to our model system where we use lysozyme, hen egg lysozyme, here abbreviated as HEL. and looked at the interaction with aluminum hydroxide. So both are positively charged at the neutral pH, so lysozyme doesn't adsorb to aluminum hydroxide. Next. Advantage, yeah, thank you. And so we, that's fine. We added a linker to this lysozyme with a phosphate group. Actually, it's a phosphonate group with three oxygen molecules. And lysozyme is a small molecule, about 14 to 15 kilodalton. And so on average, we added 2 of these linkers per lysozyme to this to the protein, and then this idea that would then allow the absorption of the protein to the aluminum hydroxide. Basically, you tether, in a sense, the lysozyme, because there's an electrostatic repulsion, but because of the strong affinity of the phosphate for the aluminum hydroxide, you get binding of the lysozyme to the aluminum hydroxide. And so next slide. Yeah, thanks. So we then showed that indeed allows the absorption of this phosphorylated form of the lysozyme to the aluminum hydroxide, shown here. Next slide. And so the question again is, so does it affect the immune response? And here we immunized mice with either the native form of the lysozyme, so HEL, or with the phosphorylated form of the lysozyme, the HELP, and then at different doses. So 10 microgram per dose, 3 microgram per dose, 1 or 0.3 micrograms per dose. And then you'll see here with the antibody titers on the y-axis, at the high dose, there is a slight effect perhaps of the phosphorylation, but it's not a significant effect. But if you lower the dose, you see that there's an increasing difference between the phosphorylated form and the non-phosphorylated form of the lysozyme. And in fact, the antibody titer that we achieved with the phosphorylated form of the lysozyme at 1 microgram per dose is very similar to that at 10 microgram per dose of the non-phosphorylated form, indicating that you have a tenfold dose sparing effect because of the phosphorylation and the absorption. So absorption again can be important, especially here at lower doses of the antigen. Next slide. And Daryl Irvine's group at MIT have done similar studies. They actually did a more targeted linkage of antigens by introducing a terminal cysteine group, and then they used a couple of phosphoserines to again take advantage of the phosphate groups here that can bind to aluminum hydroxide. And you can see that with 4 of these phosphoserines, you get significant absorption of this particular antigen. In this case, it was cytochrome C, which, like lysozyme, has a high isoelectric point, so it has the same charge as aluminum hydroxide. It doesn't bind to aluminum hydroxide as shown here. But then you add these phosphate groups with the linker, you get a significant adsorption, and they went on to do studies with other antigens and showed that this also has a beneficial effect on the immune response. Next slide. So how does absorption then affect the antigen stability? We know that proteins adsorb to a solid surface, and so similar to what they do is when they adsorb to an aluminum particle, they tend to undergo denaturation. The proteins tend to unfold, and as a result, undergo denaturation. You get potentially exposure of hydrophobic residues that are internal in the intact protein, which may result in hydrophobic interactions with the aluminum adjuvants. And because there's more protein adjuvant interactions when the protein unfolds, you can get an increase of the adsorptive strength. But in the process, you may also lose neutralizing epitopes that are conformational epitopes as when the protein unfolds. So there's a potential negative effect of adsorption on the stability and the immune response. In addition, there are a number of chemical processes that can degrade proteins, such as deamidation, oxidation, and hydrolysis, and these processes are pH dependent. And so it's important to recognize that the pH to which antigens that are adsorbed to an adjuvant, that the pH to which they are exposed is different from that's in the bulk solution, in the overall vaccine solution. I'll show it on the next slide, please. So we refer to this as the microenvironment pH. And this comes back to colloidal chemistry. That's when you have a solid surface that is positively or negatively charged, it attracts counter ions. So when you have a positively charged aluminum hydroxide, it will attract hydroxyls from the aqueous medium. And that will then increase the pH at a short distance to the surface. And so proteins that are adsorbed to the aluminum hydroxide will be exposed to a higher pH than what's in the overall vaccine formulation. And so you have to keep that in mind when you do studies that examine the effect of the pH on the stability of the proteins and tweak the overall pH of the vaccine formulation to adjust for this microenvironment pH. Next slide. And so adsorption can have negative effects by destabilizing the protein, by causing chemical degradation, and just tight adsorption can also have a negative effect on the immune response, possibly because it doesn't allow the antigen to dissociate from the adjuvant after it's internalized by antigen-presenting cells, and so it may interfere with the antigen presentation. And so we've done a study here with, in collaboration with people at Merck, where we used this hepatitis B surface antigen. This is produced as a phospholipid nanoparticle in which the antigen is inserted. It has an overall isoelectric point of around 4.5, so it adsorbs by electrostatic adsorption, but because of the phospholipids, there's also evidence for ligand exchange adsorption. And so with aluminum hydroxide, which it's surface covered with surface hydroxyls, there is extensive opportunity for ligand exchange. And so aluminum hydroxide very tightly adsorbs the hepatitis B surface antigen, as shown here by the adsorptive coefficient, which is an indication of the adsorptive strength. If you treat the aluminum hydroxide with phosphates, then you lower the adsorptive strength, you lower the adsorptive coefficient. Next slide. Next picture. Yeah, thanks. And so what happens is, so you get aluminum hydroxide, we get you have a high adsorptive coefficient, high adsorptive strength, and you get a relatively low antibody titer when you inject mice with this formulation. But when you treat the aluminum hydroxide with phosphate, this phosphate-treated form of aluminum hydroxide induced a better immune response, significantly better. There was not much of a difference between the different degrees of phosphorylation, but the phosphorylation itself induced a better immune response than with the aluminum hydroxide adjuvant because of this effect on the binding strength. Next slide. So just a bit of a sidestep here. Just want to talk for the next two slides about the preparation of multivalent vaccines. So multivalent vaccines are vaccines that have multiple antigens. And you can prepare them in different ways of preparing those. One way is to add antigen A to an adjuvant, then antigen B, antigen C, antigen D, so a sequential adsorption. Next way by which they can be made is competitive adsorption. Next Next slide. Is competitive adsorption, where you combine the aluminum hydroxide or aluminum phosphate with all antigens at the same time. And then probably the best way to make them is a separate adsorption process, where you adsorb, next slide, where you adsorb each individual antigen to an aluminum hydroxide and then combine them. The question, of course, is then, so does this introduce heterogeneity in your vaccine formulation? Because you now have particles that have aluminum, that have adjuvant A, particles that have adjuvant C, and etc. Well, that turns out not to be the case. Next slide. From the left here, the labeled BSA, so both on serum albumin with a red fluorescent marker and then with a green fluorescent marker, and then adsorb each independent to aluminum hydroxide adjuvant, and then combine them and mix them. When we do that over time, you see that there's deaggregation and reaggregation of these adjuvants. So again, remember that these adjuvants consist of nanoparticles that form these loose aggregates. And though there's the continuous deaggregation and reaggregation when you mix these adjuvants. And so over time, there is it introduced more uniformity. So even if you adsorb them separately, and before you mix these antigens, over time you get particles that have all four antigens at the cell surface. And then Donatella Lera, with his colleagues at GSK in Siena, and Derek was involved in the study. They did a similar study where they adsorbed antigens A, B, C, and D, had monoclonal antibodies against these antigens, and then looked at the distribution of these antigens over the aluminum particles by flow cytometry. And if you do this by separate adsorption, you can see that there's individual particles here that have only antigen A, only antigen B, only antigen C, and only antigen D. When you do sequential and competitive adsorption, the particles have all four antigens together. But over time, when you incubate these adjuvants, these vaccines, they use up to 100 days or 130 days, then the distribution becomes homogeneous regardless of the process by which you make these vaccines. Next slide. Now we'll finish up with talking about the mechanisms by which aluminum adjuvants enhance the immune response. And I'll talk a little bit about the depot effect, the effect that adsorption has on the uptake of antigens, and then the role of inflammation at the injection site in the response to aluminum adjuvanted vaccines. Next slide. So I mentioned that antigens that are adsorbed via electrostatic mechanisms rapidly desorb. And so ovalbumin, for example, is negatively charged, so it adsorbs to aluminum hydroxide electrostatically. But then you put it in lymph fluid, so to mimic what happens at the injection site, it rapidly desorbs. Nevertheless, next slide. When you examine the presence of the antigen, in this case ovalbumin labeled with a fluorescent fluorochrome, you can see that the mouse on the left here received the soluble ovalbumin, the one on the right received ovalbumin adsorbed to aluminum hydroxide. Over time, there is more of the antigen is retained at the injection site. So there's clear evidence of a depot effect, even though it's short lasting, not many days. Next slide. So the question, of course, is that depot required? A much cited paper from James Brewer's group in Glasgow showed that the antigen depot may not be required for the adjuvant effect of aluminum adjuvants. But they used a system where they injected a combination of aluminum hydroxide and ovalbumin into the ear of the mice, the earlobe of the mice. And then they removed the earlobe, resected the earlobe after several hours or days to look at the effect of the immune response. It's so-called Van Gogh experiment, named after the Dutch painter. And so what you see here is that even after 2 hours, if you remove the injection site within 2 hours after injection, there was no effect on the immune response. However, it is important to keep in mind, so the evidence here presented was very clear, but it's important to recognize that the earlobe is a very unique way of injecting, and it doesn't mimic the injection in muscle or subcutaneously that we typically do with aluminum adjuvant vaccines. And it's a very tight tissue, and so whatever you inject is squeezed out very quickly. And in fact, Stephanie Eisenbarth from Yale, in a review on dendritic cells, also mentions this, that as you can see here, that depending on the site of the formulation, the hydrostatic pressure in the ear lobe can force the antigen delivery to the draining lymph nodes within minutes. And so this is really an artificial system. Next slide. And so Dennis Christensen and his colleagues in Copenhagen, they did a study more recently where they looked at the immune response to a chlamydial vaccine antigen, CTH522, formulated with 3 different adjuvants, including aluminum hydroxide. And then they injected the combination and got an immune response here after several weeks. But then if you remove the injection site after 6 hours, 24 hours, 72 hours, you can see that even after 72 hours, the immune response is negatively, with aluminum hydroxide, is negatively affected, indicating that the depot effect is important. And I think we have now more evidence that the slow release of antigens from the injection site can actually stimulate the immune response and is beneficial to the immune response. In addition, the adjuvants cause inflammation at the injection site, which is important for the recruitment of antigen-presenting cells. In particular, muscle, skeletal muscle, has very few macrophages, very few dendritic cells, and so you need some time to recruit the antigen-presenting cells into the muscle through an inflammatory process. Next slide. And so if you inject an aluminum adjuvant, adjuvant is vaccine into the muscle, and here the adjuvant is labeled with maurine hydrates, giving a green color, and then you have here the muscle fibers, and then you see after 1 day you have already inflammatory cells. There's some edema. After 7 days, there are macrophages which have gobbled up most of the adjuvant. But then importantly, on the right here, you have the cells that are monocytes. They're labeled for MAC2 or galectin-3, and here for MHC class 2. These are the cells that are infiltrating the tissue, differentiate into dendritic cells, and then pick up the antigen and the adjuvant, shown here in green inside the cell, and then take that to the lymph node. So the depot effect helps to retain the antigen at the injection site for some time to allow the influx of inflammatory cells and the uptake then by inflammatory cells or by the dendritic cells to take them to the lymph node. Next slide. So here on the left, show that when you absorb an antigen to an aluminum adjuvant, you increase the uptake of the antigen through phagocytosis. So it's a more efficient process than just taking up soluble antigens. And on the right, we showed here that the effect of adjuvants, aluminum adjuvants on antigen presentation to incubated dendritic cells with ovalbumin, soluble ovalbumin, or ovalbumin mixed with aluminum phosphate, aluminum hydroxide adjuvant. Aluminum hydroxide absorbs ovalbumin well, and you get significant antigen presentation of the ovalbumin peptides to T hybridoma cells. Next slide. In addition, aluminum adjuvants directly activate dendritic cells and antigen-presenting cells by inducing the secretion or the release, I should say, of IL-1 beta. And it's selective for IL-1 beta. There's no production of IL-6 or IL-12, but certainly aluminum adjuvant induce IL-1 beta release from dendritic cells. The next slide. And you can see that here for THP1 cells. So these are human monocyte-like cells that were differentiated to macrophages by treatment with PMA and then primed with LPS. And if you add aluminum phosphate, you can get a significant release of IL-1 beta. And that's inhibited by MCC950, an inhibitor of the NLP3 inflammasome, by this compound CA074, an inhibitor of catepsins, and by cytoglycan D, an inhibitor of phagocytosis. And based on this, and certainly a lot of work from other investigators, we believe that now the mechanisms by which aluminum adjuvants induce IL-1 beta release is by activating the NLP3 inflammasome. So next. In antigen-presenting cells, the IL-1 beta is initially produced as an inactive proform that needs to be cleaved by caspase-1 into an active form. IL-1 beta that is then released from the cell. The caspase-1 is associated with the NLP3 inflammasome. So adjuvants, like aluminum adjuvants, are taken up by antigen-presenting cells that is inhibited by cytoglycan D, as shown here. That results then in endosomes that are destabilized by the adjuvants, that result in the release of catepsins, so proteases. That the exact mechanisms are not quite understood yet, but that somehow really cause the activation of the NLP3 inflammasome that is inhibited by MCC950, and so in the absence of the inhibition of the inflammasome results in blockage of the IL-1 beta release. Next slide. I also want to mention here that aluminum adjuvants have been shown to activate the complement system. A study here by a group in Denmark that showed that when you incubate human serum with adjuvant, with an aluminum hydroxide adjuvant, you get a very rapid release, production of C3A, to less extent C4A, but also C5A. And C3A and C5A are known as anaphylatoxins, so molecules that drive inflammation and can also directly activate antigen-presenting cells. And I think this is a probably somewhat underappreciated aspect of aluminum adjuvants. Next slide. So this sort of tries to summarize the ways by which aluminum adjuvants can activate the immune response, especially focused on the dendritic cells. And this is based on work from a variety of research groups over the years. So you can see that the aluminum adjuvants, they can directly activate dendritic cells. They can increase the uptake of antigens and increase antigen presentation. They can activate the NLP3 inflammasome. They can do it indirectly by inducing some cell damage at the injection site, which causes release of these danger-associated molecular patterns like uric acid, ATP, and DNA, which activate dendritic cells. And then there's also complement activation. This activation of the NLP3 inflammasome results in the release of IL-1 beta, which then activates and helps to activate and differentiate CD4 T cells and also induces inflammation. I should say that so there are multiple mechanisms that are involved. It has been difficult to demonstrate the importance of each of these individual components in the in vivo immune response induced by aluminum adjuvants, and so there's clearly some redundancy in the system, but certainly there's no question that all these mechanisms play a role in the way aluminum adjuvants activate the immune response. Next slide. So in conclusion, then, aluminum hydroxide and aluminum phosphate are very different adjuvants. They are aluminum adjuvants, but they have very distinct physical and chemical properties, which affects how they interact with antigens or other immunostimulatory molecules. The absorption can be manipulated, and it depends on the buffer, on the pH of the buffer. And then adjuvants can enhance the immune response via multiple, probably redundant mechanisms. Next slide. So I'd like to acknowledge the person who inspired my interest in aluminum adjuvants, Stanley Hem. We started to collaborate. He's also a professor in pharmacy here at Purdue University. He was a great colleague and really pioneered some of the interactions between antigens and adjuvants from a physical and chemical perspective. And when we started to collaborate, I sort of brought in more of the biology and immunobiology to that work, and we had a number of very talented postdocs and grad students working with us. I'm continuing to do this work with personal work with aluminum adjuvants. I think there's still more to be discovered, but certainly Stan was an inspiration to many of us, I think, here also on this workshop. So thank you very much for your attention. Happy to, I think I'm probably a little over time, but happy to answer questions later in this workshop.
Thank you, Harm. This was a really nice talk. Yes, we'll. If people have questions, please add them to the chat, and we'll move to the next speaker. That's Lauren from CRODA. Laura, can you get the slides up back again?
I'm going to in 1 second. I'm just trying to fix the issues we were having.
Okay, thank you.
I don't know if anybody's hearing me, but I'm still missing the presentation.
Yes, I think Laura's trying to fix.
Yeah, okay, thank you very much. Because I saw the take control button popping up. Well, I'll just present myself whilst Laura is trying to reestablish the presentation. So I'm Laurent Duroux. I'm actually lead research scientist at CRODA Denmark. In charge of working in the R&D department. I'm actually a biochemist from training, and I've been working for CRODA Denmark for the last 10 years almost, in charge of development of new adjuvant systems, and also continuing the journey with aluminum adjuvants. And that's the reason I'm there today, as if you want, as the specialist in CRODA Pharma about this type of adjuvant system. So in this talk, so thank you first to Harm having presented before me what are aluminum adjuvants. I think you had now a good overview. I'm not an immunologist, so you will hear very little from me about immunology. Just a couple of points to mention a number of things, but that will be it. I will focus today on really the chemistry and the production of these aluminum adjuvants. So in this talk, after a quick presentation of who we are at CRODA Pharma and what we do at our manufacturing site in Denmark, I will tell you about the manufacturing of amorphous aluminum hydroxide phases under CGMP, used as adjuvant systems in commercial vaccines, both pharmaceutical and veterinary. But I will, as I said, I will set the focus on the chemistry of aluminum hydroxides to make it clear that these materials are complex and potentially heterogeneous in nature. So, in fact, the existential challenge for us to produce aluminum hydroxide is to harness this potential structural heterogeneity, such as the resulting products, which are, in our case, Alhydrogel and Adjuvants, products that you have probably used already, have indeed a consistent and defined characteristics and properties, and to behave as expected in the hundreds of millions of doses of vaccines administered every year. So just okay, might have a problem to change the slide. Laura, if you can hear me.
Can you see your take control function again?
I think
it was
you're right. I missed that. Yeah, sure. So I'll press control. There you go. Okay. So thank you for your patience. So first, as I said, who are we? Where are we located, and what do we do? So we are, in fact, Croda Pharma, and we are located in Denmark. So Croda Pharma is effectively the name of the division in charge of the production of pharmaceutical products at Croda. And here in Denmark, we are dedicated to the production of aluminum adjuvant systems. And we are located in Denmark, as I said, close to Copenhagen, about 50 kilometers northwest of Copenhagen, where we have our manufacturing site for the aluminum adjuvants, but also for the saponins, which I won't talk about today, and also our warehouse, which is also located nearby. Recently, at the beginning of 23, we relocated the R&D group team, who's led by Dennis Christensen, to the Danish Technical University campus, over a year and a half ago. So if you want to come and visit, I would recommend to come in spring, which is the best year. But of course, we'll take visitors all year round. So, probably our adjuvant systems. Croda Pharma is not a manufacturer of vaccines. It's a manufacturer of chemicals, specialty chemicals for the manufacture of different type of pharmaceutical and APIs compounds, also excipients. And in fact, through the acquisition over the years of a number of companies, it has effectively built a repertoire of specialty chemicals for this particular purpose. So if we focus on at the site, as you might know, in fact, the site has got an history, which is stemming from the first manufacture plant for aluminum hydroxide or oxyhydroxide created in 1939 and operating in the premises of Superforce, which was a large chemical company in Denmark. Then over the years, this manufacturer has migrated to the actual site. So that was in 90. And this was piloted by Erik Lindblad, that you might have known. And more recently, this site was acquired. So I will just... sorry. Somebody's talking. Okay, thank you. And the site has been acquired by Brenntag, and that was the previous owner. So in 2000, and more recently, so by Croda in 2018. And since then, Croda has acquired a number of other companies to complete, if you want, the picture in terms of aluminum adjuvants and immunostimulators. In particular, Avanti Polar Lipids, who are manufacturing, for example, FAD products, which are lipid TLR4 agonists. So I won't develop on this. Croda has got a global presence, international presence. So this slide is a bit outdated, but just to give you an idea, it's present in 37 countries, has got 85 operating sites, and employs now well over 6,000 employees, and is now structured in different divisions. And the division I'm actually working for is, as I mentioned, Croda Pharma, with 5 different sites manufacturing adjuvants systems or immunostimulators. So you have to be aware that, sorry, it went a bit too fast, that our manufacturing site in Frederiksund are both the plant and the production facilities are certified CGMP by the Danish medicine agencies since 2020. And this regulatory agency is performing, of course, regular inspections, which are conducted to check in on the compliance of the site to CGMP rules. Starting with this accreditation by the authorities, Croda Denmark has been under already the previous owner, Brenntag, investing into the development of the site. Sorry, somebody is talking again. Could you mute your PC? Thank you. So Croda has been, or let's say the previous owner, Brenntag, has been investing quite massively to grow the capacities. And this growth has been sustained and accelerated by after the acquisition by Croda in 2018. And in particular, to upgrade some of the equipment for the production, and also to build new buildings for pipeline adjuvant systems that we are currently developing. This capacity is, if you want, materialized in the form of larger sterilization vessels and storage tanks to increase the capacity of production for Alidogel and Adjuvants. The implementation of water for injection purification plant, and also a new centrifuge line for the production of Alidogel. And in 2021, the management has decided to also create a dedicated process development team to sustain developments in the new pipeline adjuvants. So here you will see now just a few pictures to give you, if you want, a feeling for the plant with new centrifuge suits, ultrafiltration systems for let's say the deionization of Alidogel, or also saponins, and ultrafiltration and freeze drying for the saponin part. So it's not concerning really aluminum hydroxides. And here another series of pictures to show you that Croda Pharma aluminum adjuvants are guaranteed sterile, which of course necessitates packaging in clean rooms under very strict conditions. And in fact, this packaging is done under aseptic conditions in grade or class A or B facilities. So for this to ensure the highest quality and safety profile for our adjuvants. Now talking about the products which are in fact manufactured at the site. So as you know, it concerns essentially the aluminum hydroxides, which are hydrogel and aluminum hydroxyphosphate, Adjuvants. But the site is also responsible for the production of saponins, quillae, and QS21, which of course I won't develop here. So every product comes in particular packaging. Aluminum oxyhydroxide and aluminum hydroxyphosphate are phases which are dispersed in water, so it comes as a suspension. These are some of the best studied adjuvants and have over several decades of safe history in commercial vaccines and also clinical trials. There is, as I mentioned, a certified production for aseptically prepared steroid products, which is accredited by the Danish authorities under CGMP. And these products, in particular, hydrogel, come in different variants in terms of concentration. So there is effectively hydrogel 2%, 1.5%, 1.3%, and the variant hydrogel 85 and 7 wash for a specific customer. And these products are packaged in polyethylene containers ranging from 250 milliliters to 25 kilograms. So this is for the general, if you want, logistics. And these adjuvants are, as I indicated already, used in human and veterinary vaccines altogether. So now I will just raise the question is what is expected from alums? So by alum, I mean specifically the products of precipitation of aluminum ions by complex formation with a base. So we are referring to effectively the product and not, as Harm mentioned, to the sulfate salt of aluminum. As Harm mentioned as well, there are essentially three forms, which are aluminum oxyhydroxide, which we call hydrogel, aluminum hydroxyphosphate, which we call Adjuvants, and there's a third form, which is essentially the property of Merck, which is aluminum hydroxyphosphate sulfate. And these systems are essentially function. So they are essentially used as adjuvant systems to both induce a desired immune response and also for protein antigen adsorption. So I won't get through this because you have had an extensive presentation from Harm. But what counts for us is definitely to produce materials with the highest batch-to-batch consistency. And this, as you will see, can be a bit of a challenge in itself. So I will talk today essentially about aluminum hydroxides, precipitates, and their polymorphism. And as you will understand rapidly, it's all about the chemistry. So in production, we are essentially, or the colleagues are essentially concerned with the process parameters and the different, what happens at different stages. So that's what I will guide you through today, is to have a deeper look into these different stages and what we know about these different products and how does it impact, in fact, the qualities of the final product, which is aerogel. So in principle, the manufacture of aluminum adjuvants is a simple chemical reaction, but it contains it has its own complexities, which I will guide you through. So starting from, let's say, a salt of aluminum in dehydrated form, for example, aluminum chloride. Well, the first step concerns effectively solubilization of these ions in aqueous systems or water, to take it simply. And as soon as these aluminum ions are effectively solubilized, they will form a complex with water. So it's actually an acid-base reaction based on the reactivity of aluminium, which is a strong Lewis acid. And this aluminium will react with bases to form a complex. And as water molecules are around, that would be the first base that aluminium will react with to form effectively an hydrated form, which is, for example, shown there in blue. And then depending on the type of base which is used, so you can see that they can be strong bases such as sodium hydroxide, potassium hydroxide, or calcium dihydroxide, or eventually also ammoniac. If aluminum is effectively put in presence of these bases, well, you will get essentially aluminum hydroxide or oxyhydroxide. Whereas if you use the conjugated base of phosphoric acid, so for example, 3 sodium phosphate, you will get now a complexation with phosphate ions, as Harm has indicated, to get aluminum hydroxyphosphate. So all of this looks very simple, and but in fact it's more complicated, as I will show you. The good thing is that these systems have been effectively studied for many years, and if I have to mention one particular publication, that's probably again a publication from Stanley Ham's group, the publication by Shirodkar in 90, who already identified the phases of aluminum hydroxides in this seminal paper. And using X-ray crystallography or diffraction. So I will come back to that. But in fact, this paper kicked off decades of research on aluminum hydroxide manufacture and their physical and chemical knowledge in the field of vaccine adjuvants. And there were already some structural elements by X-ray diffraction showing some variability in the different phases, meaning whether it was aluminum oxyhydroxide or aluminum phosphate materials. So I would like to pay my tribute also to Stanley Ham for the massive work he has done in this field. Now, looking at the process itself, I hope sorry, I clicked twice, and then it goes a bit too fast because I'm impatient. Laura, could you put back the previous slide? I'm sorry. Yes. So just to give you an overview, a very simplified scheme for the production of Adjuvants, because this is I would say the simplest system. Yep. I've lost the presentation. Yeah, thank you. Where you can see that this process can be divided in 5 steps: dissolution, precipitation, and purification and concentration of the precipitates, heat fixation and adjustment in the case of Adjuvants, autoclave sterilization, and then the final filling of the product. And starting with, as I mentioned, aluminum salts, which can be of different type, and of course the base. If it's alylogel, then it's going to be, for example, a strong base such as sodium hydroxide. If it's If it's Adjuvants or aluminum hydroxyphosphate, it would be, as I mentioned, phosphate ions, basic. So I will now guide you through the different steps to understand. So my point today is to make you understand what's happening, what is the different if you want, chemical steps in the process, and in the final product that we get. And also to put an emphasis on the fact that hydrogel or Adjuvants are effectively complex products because they are amorphous and they can have effectively different phases, which can have, of course, an impact on the material properties in the end. Take control. I've got to take control. Sorry. Request control. Okay. Okay. So step number one is what happens when anhydrous aluminum salts are dissolved into water. There are various salts of aluminum, the most commonly used being the double salt aluminum phosphate or aluminum, sorry, potassium sulfate. And as soon as the aluminum ion is put in water, it instantly reacts with water molecules to yield an hexavalent complex with water molecules. And that's the form there on the left that you can see, where you have effectively an aluminum ion, which is hexa coordinated with 6 molecules of waters. So these are coordinated oxygen atoms through a so-called dative covalent bond. So you can consider this type of bonding as covalent to a central aluminum ion. And then you can see already the geometry of this particular ion complex is bipyramidal or octahedral, where you have effectively 8 edges. In this particular form, it forms effectively a complex with 6 water molecules, and then is formally positively charged with 3 positive charges. But as soon as this is effectively happening, there will be a series of equilibria which will be established by reaction with water molecules, where one water molecule will eventually abstract 1 proton from one of the water molecules coordinated with the aluminum into a so-called hydrolysis reaction, and to yield different forms, so different let's say, different complexes of aluminum, each with a different formal charge, +3, +2, +1, 0, and eventually -1. And all these ions do have a different solubility. So as you understand, in a pH range, so what we're interested in is having effectively conditions where there is effectively a mass transfer of aluminum from the solubilized phase to the solid phase, so as a form of precipitate. And that's if you want, the phase that I represent there in red at the bottom, which is the neutral phase, which is aluminum trihydroxide, also complex with 3 water molecules. So in this case, this material has got a neutral charge, but has got also the lowest solubility, as I will show you in the next slide. So in fact, what we are doing is starting from a solution of aluminum, which is very acidic, by adding a base. We put effectively these series of equations that you have seen, such as to get essentially aluminum trihydroxide, which is the neutral form, which has the lowest solubility. And in this particular case, the material will then go from a sol gel phase, so a gel form which is homogeneously dispersed, into a solid phase which is amorphous, which is then precipitate. And so in fact, at the concentration of aluminum which is used in the manufacturing process, which is well beyond 10 to the minus 1 molar. So if we look at the log concentration there on this graph, you can see that 10 to the minus 1, you are up there. At this concentration, there is a large excess of aluminum ions. And if you look at the solubility profile of the different ions, you will see that in the pH range from 6 to 8, this is where aluminum complexes have got the lowest solubility and therefore precipitate. So the process simply consists in alkalinizing the solution, such as to reach this pH range. Now, the result of this is that one gets effectively an amorphous phase of aluminum hydroxide, but this amorphous phase is not stable and will, with different parameters, evolve into crystalline phase such as gibbsite, boehmite, and biarite. Again, Harm has mentioned some of these, and in our case, we know that aluminum oxyhydroxide used as adjuvant is a boehmite phase, so that's essentially our target. What we want is to operate or to have process parameters which guarantee that the pH of precipitation is comprised between 6 and 8 to get essentially the boehmite phase. And I've just represented there the structure of gibbsite, which can be, if one doesn't pay attention to these process parameters, another phase which can be formed at a more acidic pH, and which consists of layers of crystalline lattices like this, organized in different layers. And between the lattices, there is effectively hydrogen bonding, which holds these lattices stacked together. And that's what results in the picture that you have seen already just before of these hexagonal shaped platelets, which effectively are crystalline gibbsite phase and which are not used as adjuvant systems. So if you want to summarize or to see it under another angle, indeed, if you precipitate aluminum ions with changing the pH, you can get polymorphism in terms of phases, and depending on the pH of precipitation. So as I mentioned, if the pH of precipitation is low, between 5 and 6 or below, you essentially get a material which evolves to gibbsite as a solid phase. If the pH is above 8, then you will get another type of phase, which is biarite. And the phase that we want to get is obtained when the pH range is between 7 and 8, and which is called boehmite. So these different phases, crystalline phases, which are forming as a function of time, also depend on the temperature. So I won't get into the details, but for getting, as you can see actually on the graph, you can see that, for example, boehmite is typically formed between 7 and 8 at a slightly elevated temperature, around 60 degrees. All these phases can evolve to a so-called metastable gamma alumina phase, which is then characterized by a particular structure. And these different phases can be identified by their X-ray diffraction patterns, as you see on the right there, where the two central phases there, which were obtained at pH 7 and 8, corresponds to the profile of boehmite, whereas the phase obtained at pH 5 is gibbsite, and the phase obtained at pH 11 is biorite. The shape also of the peaks will indicate something about the crystallinity or the amorphous composition of the phase. So this is very interesting and informative information to differentiate the different phases, and also to have a form of measure of their crystallinity. So now you have understood that what happens when this solubilize and when you effectively let aluminum hydroxide or aluminum hydrated complexes to interact with each other. There is effectively a first phase where these octahedral complexes between aluminum and water self-organize into an amorphous gel sol phase, where the material is homogeneously dispersed. And as a function of time or as a function of the temperature, these phases will get more crystalline to either give aluminum trioxide-like phase, which is more like gibbsite, or aluminum oxyhydroxide like phase, which is the Boehmite phase. And if you push this reaction further, so it's a so-called in the case of Boehmite, it's so-called dehydration condensation reaction. You will get eventually gamma alumina, which is a highly crystalline phase. Of course, these materials are not used in vaccine adjuvants, or as vaccine adjuvant, but they can be effectively formed if, again, the process parameters are not well controlled. So that's something to always keep in mind to avoid effectively the formation of undesired phases, knowing that halide hydrogel and aluminum oxyhydroxide for aluminum adjuvants are defined as Boehmite phases. So now, in this heat fixation process, in a way, this is the necessary energy in the form of heat, which needs to be brought to the sol gel to make this phase transition to a more crystalline phase, where you have the formation of small crystallites, which are not completely organized into a crystal phase, but at the same time to avoid that these morph into condensed phases, such as full developed crystals. So the whole trick is to effectively bring enough heat for enough amount of time, such as in a way this aluminum oxyhydroxide phase, which is Boehmite phase, is frozen into a particular form that you will see a bit later. And as you have never understood, is because there is effectively the possibility of having different phases forming on top of Boehmite, which is our target, such as biorite and gibbsite. There is effectively the possibility of a contamination, if you want, in the final product. So this is, for example, why it's very important to control the pH, because these different phases are mostly depending on the initial pH and of the reaction, and of course the final pH. So the question is the potential for formation of gibbsite and biorite in the production process a concern? Well, luckily, the kinetics of formation of these three phases was studied by Kim and Lee. That's the figure that you see on the right there, and show that the Boehmite phase is preferentially formed up to 150 degrees, above which the other forms can dominate. And of course, in our process, we do not reach these temperatures. So we have already by nature, if you want, a situation where the formation of Boehmite phase is preferred. In another study by Baccarella, so that's the figure on the left there, show that the formation of the more condensed phase of gamma aluminate from the initial amorphous aluminum hydroxide gel is a fast process. You can see that from this UV absorption spectrum in the hard UV, so that's below 180 nanometers, that there is effectively a very fast formation of the gamma alumina in about 10 minutes. But this is at 200 degrees Celsius. And although it's not a major concern that there is effectively this more highly condensed phase of gamma alumina, which is formed in the process, there is still the possibility that some of it is formed. But globally, the kinetic factors are at play. That's clear from these two graphs, meaning the formation of boehmite or the boehmite phase or pseudo-boehmite phase, as I will show you just afterwards, is effectively preferred and favored. But these kinetic factors can influence heavily the outcome of the production and therefore the quality of the final product. What you don't want to have is effectively in our hydrogel a mixture of pseudoboehmites and then eventually amorphous forms of gibbsite and biarite, which have very different, if you want, conformation and therefore properties. So this is why effectively the process parameters have got to be very tightly controlled. So at the heat fixation step, which is the third step in the process, where we almost have hydrogel or aluminum oxyhydroxide. And what is it at this point? Well, it can be defined as a metastable pseudo-boehmite phase, which is kinetically trapped. So that sounds a bit, strange, but what it means is that that's a material which is effectively, as you can see on the electron microscope micrograph there, formed as also harm presented by nano sized crystallites of boehmite phase, which are forming aggregates of about 100 nanometers and above, and then flocks of aggregates, where these aggregates can then flock into much larger structures. You can see there on the other micrograph about 2 microns. And these flocks of aggregates are captured typically by methods using laser light diffraction, which gives effectively the particle size distribution of these materials. What is very important to understand is that this phase of pseudoboehmite is kinetically trapped, meaning that it's arrested in a particular pathway, a chemical pathway, where it still evolves into eventually the final form, which will be gamma alumina, which is a much more condensed form. And we'll see that this has effectively an importance when, for example, the material needs to be post-processed or handled. I just wanted to show you also on the right there an X-ray diffraction pattern of obtained by Bocchini or published by Bocchini et al. in 2001, of this transition between the salt gel initial aluminum hydroxide phase, which is a salt gel, into the first amorphous pseudoboehmite phases by just raising the temperature to 22 degrees, 30 degrees, 50 degrees, and then above, where you can see that by monitoring or by recording these X-ray diffraction patterns, you can effectively get information about this transition, in particular between the salt gel phase and the first, if you want, appearance of crystallites of boehmite. And this is signed by this reflection at 0 to 20, which is effectively indicative of the stacking of the crystal lattices in the boehmite phase. So this is also a very, if you want, powerful way to follow the kinetics of formation of the material. Now, the fourth step, which is the sterilization of the material by autoclavation, is nothing more than, if you want, an acceleration of this process where this condensation, dehydration of the boehmite phase will then proceed and extend such as the after autoclavation, the material is even more condensed. If you push the process to the extreme by either increasing the time or the temperature, you'll end up with, if you want, a pure bumite or bumite phase, which is represented there on the micrograph on the that would be the right, sorry, where you can see that the size of the crystallites are about 2 micrometers with regards to the size of the crystallites in our hydrogel, which are tens of nanometers in size. But that's essentially chemically the same material, just that the phase is more condensed. So you understand that the whole process of manufacturing this material is about controlling this growth. And that will determine the crystallite size, the accessible solvent area, so meaning the potential absorption sites for protein antigens, for example, and the aggregate and the flux formation in the final and the size distribution of the final product. And of course, it will also impact the sedimentation and rheological properties of hydrogel. So this was well understood again by Helm's group, who actually looked at the effect of autoclaving and repeated heat cycles on hydrogel, and who showed clearly in a publication by Burrell and Helm's group in '99 that if effectively you subject aluminium oxyhydroxide to autoclavation or aluminium hydroxyphosphate, so Adjuvants, to autoclavation, it leads to a change in acidity, a change in the points of zero charge, typically by a drop in the points of zero charge as a function of the time of autoclavation, and a reduction of the adsorption capacity for antigens. And that's even more marked for Adjuvants than it is for aluminium oxyhydroxide. And this was confirmed more recently by Yu et al. By publication 24, where they observed the same effect, of course, and also brought a bit more by looking at the sedimentation properties of the re-autoclaved hydrogel, showing that that's what you see on the extreme right there, this figure, where you can see that the sediment packing after 24 hours is much higher. So that means that the dispersed phase of hydrogel is lower than before re-autoclavation. So re-autoclaving hydrogel, I know this is often asked question by our customers, is definitely not without consequences. You have to be aware that because of the chemistry going on, the phase will be more condensed, and there will be effectively a loss of surface area, global surface area. Harm mentioned about 500 square meters per gram of material. We have measured similar values for both hydrogel and Adjuvants. Actually, Adjuvants, it's even higher. It's about 700 square meters per gram using different techniques. And this surface area is largely reduced due to the compaction of the material. To recapitulate, so I didn't show anything about Adjuvants, but in principle, it's the same. Of course, you end up with different material where now phosphate ions will establish a covalent bond with aluminium, and eventually will form an extended network of aluminium, phosphate, aluminium by binded coordination, and again form crystallites and phases which will then assemble. The big difference between Adjuvants or aluminium hydroxyphosphate and aluminium hydroxide is that the nature of the interlattices bonds is different. With hydrogel, you have hydrogen bonding. With Adjuvants, you have phosphate bonds between aluminium atoms belonging to two different phases. And that has effectively also a consequence on on the properties of the material, and they also their solubility and so on. But basically, in terms of recapitulation point, making or manufacturing amorphous aluminum hydroxide of Bumid phase will be about controlling the crystallite size and the interfacial properties of the material, surface charges, surface dielectrics, edge bonding density, surface area, crystal shapes and edges, and eventually the electrostatic potential, and combined with the crystallite size, that will control so the aggregate structure and size and the flock structure and size, which is effectively the physical properties of this material, which have a strong impact on its biological properties, and starting already with adsorption of protein antigens. So I wanted to just to exemplify this, I wanted to show you a real life experience that we have had with aerogel manufacture and production recently, which is an out of specification situation. One of the parameters that we are measuring is sedimentation, according to a very simple test, and as I mentioned, the manufacturer line has been updated, and as a consequence, the product has shown, in particular in the phase of validation of the whole new process, have shown a difference in sedimentation properties, such as then in theory it wouldn't pass the test and wouldn't be able to be released. So there I show you here if you want the sedimentation test, where you simply put a hydrogel into a graduated tube and you measure the height of the dispersed phase after 24 hours. And you can see there, normally we expect at least 90% of, if you want, the specific volume for this dispersed phase. In the case of this material failing the test, as you can see on the picture there, the dispersed phase was only occupying 50% of the total volume. So then we have investigated what could be the cause of this. Does it have to do with if you want, the electrostatic charge is measured by zeta potential, so the potential, if you want, displacement of counter ions, which could impact on the charge of the material. Essentially, the chemistry was exactly the same, the process parameters are the same, so we wouldn't expect a very different phase. But because now the centrifuges, for example, to wash all the free ions are more powerful, maybe it had to do with the displacement of or the washing of these ions, which would have then an impact on the isoelectric point or zeta potential. And indeed, we could see there are a slight difference between, let's say, the standard hydrogel, which is typically released, and then a batch which failed, for example, upon dilution. So maybe, yes, the charges have been changed as a result. Then we also looked into the size distribution. It seemed like the hydrogel batches which effectively failed this test had a higher proportion of larger flocks or aggregates than the normal hydrogel, and also the behavior of this material, this batch which failed the sedimentation, was also a bit different upon serial dilution in water. Again, which could explain this different behavior in sedimentation. Hydrogel sedimentation is a very complex phenomenon because it's a complex material. There's multiple equilibria which are in place, controlling the aggregation of the flux and so on. So I won't get into the details, but that's what essentially can be measured or picked up by the particle size distribution with laser diffraction, where one can see effectively various profiles corresponding to different subpopulations of flux or aggregates. So we also looked into the rheology, because for me, sedimentation has got to do with rheology, meaning the way these different crystallites or aggregates of crystallites or flock of crystallites interact with each other upon flow, upon shear stress. So you are well aware that when we manufacture allied gel or Adjuvants, well, there's different tanks, and the material needs obviously to be, shipped from one tank to the other through tubes. And every time you put effectively pressure to do that, you subject the material to a shear stress. And here, I was interested in looking at the response of allied gel to increase shear rate with just rotational rheometry using a coaxial system. And as expected, allied gel does effectively respond like a non-Newtonian shear thinning fluid, where the apparent shear viscosity decreases as a function of the shear rate. And based on this, when we compare the different batches of allied gel holding or failing specifications with regards to sedimentation, we couldn't see any obvious pattern, meaning that we had to look for something else. Looking at another type of experiment that you can do with rheometry, using this time oscillations, so oscillatory stress, you can get information about the elastic component in the gel if you consider that this is a viscoelastic material. So upon a certain shear stress, you will see that there is effectively an increase in the apparent shear viscosity up to a point where the material starts to flow. And you can determine a particular point, which is called a yield stress, which determines this particular shear stress, which has to be applied for the material to start to flow. Again, so what was interesting is that we could see that different batch of allied gel, irrespective of whether they hold sedimentation or not, had particular profiles. So meaning that based on this criteria, no, allied gel is not, a unique material which has a very well-defined behavior, rheological behavior under this test. And the other thing is that if you look at the curve on the bottom, this is actually allied gel which was stressed prior to the test, and which shows that if you stress allied gel with a particular, shear stress, then you get a material which has lost completely its elastic component and which starts to flow immediately. So the consequence of this is that one, allied gel and Adjuvants are materials which are extremely sensitive to shear stress. And understanding what controls this behavior is very complex. And at the same time, it doesn't seem like there's an obvious relation between this behavior and the sedimentation. So we need to do some more work to understand this. So now I'll finish with mentioning about the product quality attributes and the process control to ensure these quality attributes. And why have I talked about all of this? Aluminium oxide phase, aluminium hydroxide phase, and so on, biarrhea, gibbsite, and bumite. Does it matter in the end with regards to immunology? Well, yes, it does. And at least there's few articles which have effectively investigated this, meaning looking at the immune response of pure adjuvants, aluminium adjuvants, using model systems and looking at their effect. So I like to cite this one from Vreeling et al. in Bernard Metz group there in Intravac, who have shown that yes, they have actually synthesized gibbsite, bumite, and used hydrogel as another phase, and showed clearly that they're using PBMCs from human blood, that the immune profile was quite different, and that alum was the most effective to induce effectively an inflammatory response, and which is then also identified there on using this time cytokines profiles on cultivated THP1 cells, so macrophages, which shows that only alidogel was effectively able to induce to high levels the production of IL-6. IL-1 beta in this case. So yes, the different phases will eventually create different immune responses. So that's also something to keep in mind. Of course, our material comes with specifications with regards to aluminum content. That's very important. And then eventually contaminating free counter-ions, which were used in the process, for example, sulfate. There's also a few other parameters concerning the safety, like pyrogenicity, and also the protein adsorption capacity, which is supposed to capture, if you want, in its complexity, the material through adsorption capacity. So if we look at the specification of a hydrogel with regards to which are actually stemming from a European pharmacopoeia monograph, and which have been adapted for our particular purpose, you can see that hydrogel is effectively on the specification sheets comes the aluminum titer, the content of these sulfate, nitrogen, also phantom metals like iron and so on. The pH, adsorption capacity for proteins measured with human serum albumin, and then a test on sterility and pyrogenicity. So this is defining what is actually on the specification sheet of hydrogel. If we compare it to what is present in the original European pharmacopoeia, there's additional tests which are effectively recommended: the presence of chlorine ions, the presence of nitrates, the presence of heavy metals, arsenic, and also the sedimentation property that we don't have as on the specification, but that we measure anyhow, as I've shown you, and also the presence of endotoxins. The question is, are these specifications capturing what is really important, what we need to have for ensuring that hydrogel is effectively a consistent product? And we have seen that maybe not. What about the physical properties of the material, such as the zeta potential, the surface area of the material, the particle size, crystal flux structuration, and the rheology properties? All of this is not mentioned in the pharmacopoeia and is not done consistently or at least not part of our specifications. So there are questions to effectively consider for the future. Of course, during the process, there is effectively some parameters which are measured. I won't get in the details, and actually I'm not allowed to mention them, so I'll just pass, but be aware that for each step, there's effectively inline and offline parameters which are measured to ensure that in the end we get effectively what we want. So this can be effectively, there's very nice studies there for lately stemming from Merck and the research group in Canada, where they have implemented inline process monitoring and PAT technologies to effectively measure quantitatively and also as a function of time the formation of these different phases. They are using, for example, FTIR, where they have identified the peaks corresponding to particular, let's say, forms of aluminium or phosphate, and then to be able to monitor as a function of time the formation of aluminium hydroxyphosphate. So this is also very interesting and part of future implementations. I'll finish with mentioning something about aluminum vaccine adjuvant toxicity very rapidly because I'm a bit running after time. I will not dive into is aluminum toxic as aluminum adjuvant in vaccines. This has been debated many times. I'll just refer to a recent publication from the Dutch National Immunization Program, published by Afortit in 20, and with a very nice actually survey of all the data available in in Holland, or in the Netherlands, sorry, with regards to the total exposure to different forms of aluminium, including vaccines or vaccine adjuvants in the different class ages. And their conclusion is supporting previous surveys from WHO or US FDA and also the Polarisch Institute in Germany in 2015. And the conclusion is that the comprehensive risk assessment further supports the clinical trial and epidemiological evidence of the safety of aluminium in vaccines. I really recommend to read this because that clarifies, I think, pretty much everything with regards to safety. We're more concerned about sterility and endotoxins. So, as I said, both our aluminum adjuvants are effectively produced sterile, and but that doesn't guarantee that that there's no endotoxins presence. And despite the fact there is in the pharmacopoeia a test which is described, or protocol to test for endotoxins, this is extremely challenging because, as you probably know, endotoxins being lipopolysaccharides present effectively phosphate groups on the phospholipid part. As Harm has been effectively explained very clearly, phosphate can react to form covalent bonds to both hydrogen and NH4 through ligand exchange with hydroxyls, and therefore most of the LPS will be found, if not all, that is, in fact, will be found in the form of adsorbed. So, how to measure adsorbed LPS on hydrogen and NH4 is a technical challenge, but actually it's not even if you want, required from the authorities at the moment. But we are well aware that aluminum hydroxide and aluminum phosphate have got a huge adsorption capacity for this material, and therefore at one point it might be effectively necessary to develop tests or quantitative methods to measure this. I just wanted to mention that again from Stanley Hem, group, a publication by Shi et al. In 2001 showed that that if you effectively inject aluminum hydroxide contaminated with a particular design dose of LPS and inject it in mice and measure TNF alpha and I think it was IL-6 as well, and measure the activation or the secretion of these compounds, then there was no trace of activation of TNF alpha or IL-6 with hydrogel contaminated with LPS, but there was some for aluminum phosphate, meaning that this is mostly a concern eventually for aluminum phosphate, that it gets contaminated with LPS, as some of it can be effectively just electrostatically adsorbed and then dissolved upon injection. So in conclusion, the manufacturing of pharmaceutical grade cGMP alums is not as easy as it seems. Aluminum hydroxide and aluminum phosphates are amorphous and polymorphic, so there is a potential for heterogeneity. And on top of that, this potential can express itself post manufacturing as a function of time, or depending on how it's handled, in particular if it's re-autoclaved by our clients who are manufacturing the vaccines. The product specifications are according to European Pharmacopoeia monograph. Are they incomplete? That's a question that we're asking ourselves. The process parameters need to capture essential features from these complex materials. Again, this can be challenging, but we have seen that there's a number of techniques now which are implemented in production, in PAT, to get more information about the different phases and their kinetics of formations. And if we flip the coin around by saying that yes, aluminum hydroxides are effectively plastic into their structuration and the way they react with different bases and the way they can be effectively manipulated. Harm has mentioned about using phosphate groups to effectively covalently adsorb antigens or eventually immunostimulators. But you have to be aware that by doing some I would say, manufacturing tricks, it's also possible to manufacture aluminum hydroxide in different forms. For example, nanorods or very highly controlled shapes and so on. And that effectively gives a new potential for aluminum hydroxides to have different functions in vaccines. So I will stop there. I will thank you very much for listening to me. Yeah, just to mention that there is definitely, if you're also asking about formulation of these materials, I would heavily and warmly recommend to read the review by the three presenters today. That's Harm, Derek, and Christopher in 2018, who have given a very nice overview of these aluminum adjuvants and how they can be used and the pitfalls eventually. So in conclusion, I would like to thank you for your attention. I would like to thank the colleagues also at the CRODA site in R&D and in production. And don't forget, get yourself and your children vaccinated. And the last thing is that if aluminum kills, it's probably more in the form of a car than a vaccine. Thank you very much.
Thank you so much, that was wonderful. Um, our next speaker is Dr. Rajesh Gupta. And I don't know what this, okay, the slides are. And you can take control. Thank you, Rajesh.
Yeah. Thank you, Lakshmi. Greetings to everybody. As Lakshmi mentioned that my name is Rajesh Gupta, and I have more than 40 years experience in research, development, manufacture, quality control, and regulation of biological products, particularly vaccines. I will tell you more about myself while going through the presentation. So about the presentation, when Lakshmi asked me to make a presentation on aluminum adjuvants, I thought I have nothing new to talk about, particularly when I knew about other speakers, particularly you heard Harm and Laurent, having expertise in aluminum adjuvants. And I have never made a actually formal presentation exclusively on aluminum adjuvants, but shared my experience with aluminum adjuvants during discussing other adjuvants or delivery systems, and also with some of my clients. So in the current presentation, I would discuss certain key points that I think may be important, which I learned during my more than 40 years of professional journey with vaccines and other biologicals. So first of all, I would like to dedicate my presentation to three eminent scientists. I think everybody is thanking Professor Stanley. So that's where I started with the Professor Stanley, who brought science and sense to the dirty little secret of aluminum adjuvants. So when I started, it was used to be called a dirty little secret or art of making aluminum adjuvants. And I came to know about Professor Ham's work during early 90s when I used to get his articles for review. And when you review an article, usually you read it very carefully and spend more time. So then I learned quite a bit about aluminum adjuvants. And then when I was reviewing his book chapter in vaccine design in 1995, I had some comments that it does not talk much about the real vaccine antigens or the effect in the immune response or immunogenicity of vaccine antigens. Then the editors, Mark Newman and the other editor, asked me to write a chapter actually. That's how I ended up writing a chapter in that book also. And I also actually met Professor Ham at Wyeth when he was invited there by Lakshmi's group, I think sometime around early 2000s. And in fact, Professor Ham started at Wyeth many years back. So next one, I had the pleasure and honor to work with one of his colleagues also, Professor Harm, during 2005. I think you already heard of Harm today, and I have a lot of respect for him. We met in London and discussed about aluminum adjuvants. So about Professor Eliveld, I met him in 1987 at Leningrad, and then at the Institute Pasteur, where he took me to his manufacturing facilities in France. And I learned quite a bit about vaccines, adjuvants, and regulations from him. I consider him as my mentor. And he introduced me to numerous scientists, including Eric Lindblad from Superfast, whom I met a few years back. A few means, I think, no, maybe 20 years back, I would say. Finally, John Robbins, whom I knew from his days of FDA in early 1980s. And I met him in 1988 in India. Again, he was my mentor and teacher on vaccines, particularly polysaccharide conjugate vaccines, adjuvants, and regulations. He almost knew everything about vaccines or whatever you ask him question, he had the answer. I had the fortune of working in his lab for over 2 years during 1989 to 1991. So then I list here my publications here, because if I am not able to go into details of some of the work we have done, so that people have easy access to these publications. They are almost written 20 years back or more than 20 years back. And the one which are yellow highlighted, they are book chapters describing in details the results. If anybody wants to see the results, they can refer to the publication highlighted in green. And particularly this book chapter, like which was actually this book was edited by Dr. Hagan, describe the protocols actually how to make aluminum adjuvants. It's describe exactly precisely how much salts you take, what kind of solutions you make, how you mix them, and how you quantitate aluminum adjuvants by a wet chemistry method, how to dissolve aluminum adjuvants. If anybody interested in going into making the adjuvant at a lab scale or even at a scaled up level, probably this number 10 article may be useful. And this 11 article was the most recent. I wrote it in 2014, and it has some of the, I would say, my latest thinking on the aluminum adjuvants, that whether the aluminum adjuvants are good adjuvants for humans. I think you will see that we talk about that a little bit. And these are the three articles, as I mentioned in the three book chapters. And I will be glad to send a PDF file of any of these or other papers if anybody needs to get an easy access. So about this presentation, I would go through my professional journey focusing on aluminum adjuvants and acknowledging individuals from whom I learned about aluminum adjuvants and vaccines, and those who contributed to the work I am presenting. Then I would go over different aluminum-based adjuvants, starting with the role of alum, a few comments on manufacturing and characteristics of these adjuvants. I would share our data on role of adsorption on the potency of vaccines in animals and humans, strong adjuvanticity of aluminum compounds in animals, and their role as adjuvants in humans. A few comments on the depot formation, then moving to issues with using adjuvants as benchmark to evaluate alternate adjuvants, and finally a few comments on regulatory considerations. So I started my first job at the Central Research Institute Kasoli, a vaccine manufacturer since 1905. One of my early assignment was to set up a method to test aluminum in vaccines. I assume that there was no test done to test for aluminum content before 1978. Whatever was added was considered as content, and also aluminum phosphate was not autoclaved at that time. This was a wet chemistry method based on the method published in 1977 in a WHO manual, and I worked with Mr. Verma and Pramod Gupta. I think the method is still used actually. I was surprised in some countries as I came across this method when this was being set up at a contract testing lab in the US during tech transfer of a vaccine to the US. This contract testing lab had a lot of issues resulting in the out of specifications. So I had to go back to my 40 years to remember some of the details about the treatment of the aluminum gel with acids to release aluminum, where I boiled the solution on a Bunsen burner, and the contractor was trying to do it on a heating block set at 95°, where solution never boiled and ultimately dried, leading to not releasing all aluminum and underestimating the content. And in fact, it took a while for the contractor to get a Bunsen burner. And then there were issues with the subjectivity in recording titration endpoint. So historically, the spec used to be less than 0.85 or 1.25 for aluminium, maximum limit allowed by regulations. However, lately the regulatory agency is required to have specs based on content to monitor the consistency in manufacture. So if you look at some of the older vaccine, they will say less than 0.85 or less than 0.125. But now the regulatory agency asks to like specify the content so that they can monitor consistency in that. So my recommendation from this experience is that to set specification carefully if you are using wet chemistry method, considering inherent variability of the method and the manufacturing process, or switch to more precise atomic absorption or ICP methods. So other individuals who taught me vaccinology at CRI Kasoli include Inderjeet Sharma, Dr. Singh, Mrs. Sharma, and Mr. Mishra. So after 2 years at NIH, George Sieber asked me to work at the Massachusetts Public Health Labs in Boston, working mainly on vaccine adjuvants, delivery system, and managing clinical serology. There I had the privilege of working with eminent vaccinologists with experience in manufacturing, including Bradford Roast, actually who I co-authored a few book chapters with me, Bill Latham of the Tetrans Medium fame, and of course George Sieber, who is a great scientist and leader. Paul Griffin helped in the laboratory in the work of adjuvants. And most of the time, actually, we used aluminum adjuvants as benchmark for evaluating other adjuvants. And then we learned quite a bit about the aspects, including manufacturing of aluminum adjuvants, lyophilization of aluminum adjuvants, the isolation of aluminum adjuvants. So this picture shows the Massachusetts Public Health Lab, the manufacturing facility. At that time, now they have moved to a new facility. And my lab used to be in this building at building. This was a state laboratory institute at 8th floor. So then I moved to like Chiron for a year in Emeryville actually, and making several trips during that 1 year to formal Sclavo and Behring work facilities that were acquired by Chiron. And I consider it was a golden period to learn more from the most renowned scientists and vaccinologists, including Dr. Hagan. You will be hearing from him after me probably, Dr. Manmohan Singh, whom I actually knew from India in 1989 at the National Institute of Immunology, then Gary Van Nest and Gary Oat, who developed the MF59 adjuvant. Of course, Dan Granoff, who is also like a great vaccinologist, John Patre, Giuseppe Del Giudici, Dino Rapoli, and Martin Rears, whom I had actually met at a conference while in Boston. So in 1997, I moved to the Wyeth and got an opportunity to work with the Lakshmi's group. I think Dennis was in charge at that time with Jim Melliker, Frank Arnold. Lang Shi was at a senior position. He passed away, unfortunately, during work. And Jim Cowell, who is again a great scientist, great knowledge, worked at FDA, has a great mind in science and regulation. And of course, George Sieber again, who was here. So there I worked on various aspects of aluminum adjuvants, particularly autoclaving and CGMP compliance, which were started being implemented on vaccines in 1990s. So then I worked at the FDA CBER from 2006 to 2013. There I mainly concentrated on setting up the testing labs under the ISO 17025, reviewing BLA applications and supplements for analytical methods, and also doing some research on the rapid microbiological methods. And in between, I have been consulting and I have been currently consulting. So now to start with the aluminum adjuvants, use of aluminum containing adjuvants in vaccines started in 1920s and 30s by Glennie et al. When alum was used to partially purify protein antigens, mainly tetanus and diphtheria toxoids, by precipitating them in the presence of anions, including phosphate, sulfate, and bicarbonate ions, resulting in a mixture of compounds, mainly aluminum, sulfate phosphate, and aluminum hydroxyphosphate. Such vaccines were called alum precipitated vaccines and were used for many years. Their use declined mainly due to variability among batches during manufacture, Then there was a switch to preformed gels, mainly aluminum hydroxide, which is actually aluminum hydroxide, as you heard already, aluminum phosphate, hydroxyphosphate sulfate, which was kind of a made with the precipitation using alum. As mentioned in the last slide, use of aluminum adjuvant started with alum precipitated vaccines, but alum itself is potassium aluminum sulfate, which is soluble in water and not an adjuvant. The use of aluminum adjuvant continued as these preparations were stable. Historically, there has been no formal formulation development, particularly for adjuvanted vaccines. So any formulation work that was done with aluminum adjuvants was focused on adsorption of antigens onto the adjuvant, because it was thought that the adsorption was important. As you heard already, that there are some WHO guidance for more than 80% adsorption and FDA minimum requirements more than 75% adsorption. So there was basically no formulation. Lately, formulation has got attention, and which is important for new vaccines consisting of purified components, particularly adjuvants and combination vaccine. Attention is paid to the formulation of vaccines now. So my point here is that I think putting aluminum adjuvants or aluminum compounds in the vaccines did not need formulation. They were stable, they were good to go. As the so-called formulation of aluminum adsorbed vaccine, which was more of mixing various components, continued empirically without much understanding about the science of aluminum adjuvants, as I mentioned, which was often referred to as a dirty little secret. Art of making this adjuvant was fairly consistent within a manufacturer, with a focus on adsorption of antigens onto adjuvant. Often, phosphate buffer and other anions were used or present during formulation, which converted aluminum oxyhydroxide to aluminum hydroxyphosphate, thus avoiding binding vaccine adjuvant to aluminum through ligand exchange. You heard about it also. I will talk a little bit more about it. Degree of adsorption for same vaccine antigen, particularly diphtheria toxoid, varied considerably among various manufacturers. However, during 1990s, Professor Ahem and his colleagues brought science and knowledge to the field of aluminum adjuvants by describing chemical nature of the adjuvant and whether it is crystalline or amorphous based on ratio of aluminum to hydroxide and or phosphate, and further insight into these adjuvants by defining point of zero charge, effect of pH on point of zero charge, effects of anions, mainly phosphate ions, effect of size of gel particles, other physical characteristics which I will discuss later. For legacy vaccines, aluminum adjuvants seems to have an important role in the effectiveness of existing vaccines. Aluminum compounds has the ability to stabilize vaccine formulation, ensuring the complete delivery of vaccine antigen dose. As vaccines are used in minute amounts, microgram quantities, there is a possibility of a small but significant amount of these active ingredient lost due to protein aggregation or binding on the surface of delivery containers such as vials and syringes if these are not formulated optimally. Aluminum adjuvant provided stability to these antigens when adsorbed to or formulated with them, and additionally ensured complete delivery of small amounts of antigens. The role in helping vaccine antigen may be considered as an adjuvant effect within the broader definition of adjuvants, but this is more a formulation issue, I would say. And finally, I think previous speakers have alluded to this. Aluminum adjuvants are very versatile with regard to point of zero charge, which is discussed later also. This can be optimized to facilitate adsorption or non-adsorption as necessary, whatever you want, by changing amount of phosphate, thus changing the point of zero charge and optimizing the formulation pH. So it has been sometimes very frustrating when aluminum adjuvants have been referred as alum I have seen this in many articles actually. They don't tell anything. They said we used alum without knowing which type of aluminum adjuvant was used in the study. And as I mentioned that alum is not an adjuvant, it is soluble in water. As you can see on this slide taken from Ham and White from the 1995 vaccine design book chapter, these three aluminum adjuvants have very different physical chemical characteristics, particularly different point of zero charge. So this slide, I took it from like a Lindbland's paper in 2004. I think probably it came from Professor Ham's originally, as Harm also alluded to this study. So you can see that the aluminum hydroxide and aluminum phosphate has opposite charge at the neutral pH. And the I think Professor Harm explained this. The albumin, which has a negative charge at neutral pH, will adsorb to aluminum hydroxide, but will not adsorb to aluminum phosphate. Similarly, N-egg lysozyme will adsorb to aluminum phosphate with the opposite charge and not adsorb to aluminum hydroxide. So we heard about the aluminum manufacturing of aluminum adjuvants. As I mentioned that this book chapter actually goes into more details about how much strength solutions you have to prepare, how to mix it, and all that. And the only comments I want to make in this slide is that most common manufacturing method currently is adsorption on to autoclaved preformed gel, where aluminum hydroxyphosphate sulfate is not very common these days, as mentioned due to variability. And then there have been in situ adsorption on aluminum phosphate, particularly aluminum phosphate, was routinely done at the Massachusetts Public Health Biologics Labs for DTP group of vaccines, and can be done with the aluminum phosphate sulfate, that is alum precipitated. But however, you have to be careful that there is a possibility of ligand exchange binding, which is discussed later during in situ adsorption, depending upon saturation of aluminum with phosphate ions. So this slide actually, the data was taken from Professor Relivell's work, and this slide is actually about the calcium phosphate, which was another adjuvant actually Superfos used to make it, but they don't make it anymore. And here the message is that the quality of the gel depends upon the concentration of reactants, rate of addition of reactants, and rate of mixing. So if you mix for a shorter period and fast, you get a gel quality with less phosphate, and which has actually, this is the supernatant, the height of the supernatant. So if the height is less in 17 hours or 10 minutes, it means that the gel is not settling fast. So it is a good gel quality, small gel particles. So these conditions are also discussed in that book chapter I referred. So this again, I think repetition from what Harm presented, that for combination vaccines, you can add all vaccine antigen, mix them, and add to aluminum adjuvants, or add aluminum adjuvants to mixed vaccines. So then you can add vaccine antigen sequentially in a predetermined sequence to adjuvant, mix after every addition, or you add your individual vaccine antigens and mix. But I think he also made this point. I want to again emphasize that in the final formulation, these antigens may desorb from the gel or may adsorb more depending upon their charge. They move around depending upon their affinity to the aluminum ion, depending upon the charge actually. So this also a slide taken from Stanley Ham's work, and this is about the autoclaving. We already heard about it. I think Laurent showed similar data. And I think I just want to make a few points on this, that this can only be performed on preformed gels, cannot be performed on alum precipitated vaccines and in situ adsorption, which are manufactured aseptically, where aseptic process validation must be done, which is similar to the formulation step for vaccines. So autoclaving process should be validated. And the important point to note is that mixing of adjuvant during autoclaving is important for heat to penetrate in the center. Otherwise, heat does not penetrate in the center of the vessel. And testing it for stability does not give you enough stability assurance. If your process is not validated, you have to build the quality in the product following the principles of quality by design. As shown in the last slide, autoclaving can change the characteristics of the adjuvant, and this must be a part of approved manufacturing process. You cannot like arbitrarily say, oh, I'm going to autoclave again on receipt. And if you have to introduce another autoclaving cycle in the US, you might need a BLA supplement and demonstrate equivalency and that there is no change in the quality of the product. So these days, the quality must be built in the product by following principles of quality by design. I'm not going to go over those in this presentation due to the limited time. So in addition to the compendial tests, including stability test and endotoxin, these tests I recommend to be performed on the aluminum gel. Gel settling time in a like narrow graduated cylinder, uh, preferably 25 ml, aluminum content, point of zero charge, size of the gel, which affects absorption capacity, and also some people think that it may also, if it is less than 10 microgram, it can be taken by macrophages. Actually, this work was done by Eldridge, who worked at Wyeth, and he did this work with the PLGA microspheres. pH is important, which affects the charge on the gel. So there are some other characteristics which you can do it, but I think I will not insist that they must be done. So you can select those, and if you do it, I recommend that you do on first 5 batches to show consistency in manufacture. So these are the tests for the adjuvant vaccines. And again, I will say that you have to implement principles of quality by design, and then stability and endotoxin, and additionally aluminum content, degree of adsorption by doing dissolution of gels to release antigens, gel settling time, which changes actually when you adsorb the antigen onto the adjuvant, pH. So the other test again, I put a lot of question marks. So if somebody wants to do it, I recommend do it on first 5 batches. So I want to make a few comments on freezing and lyophilization, which is not recommended or done, which causes collapse of gel leading to aggregation and precipitation of aluminum adjuvants. However, frozen tetanus toxoid with collapsed gel was shown to be immunogenic in humans, but this is not clinically acceptable and would not be accepted by regulatory agencies. However, freezing and lyophilization is possible. Lyophilized vaccines are not commercially available because there seems to be no need or market for such vaccines. Lyophilized standards with guinea pig serum, hemosil, and sucrose have been used for more than 50 years. At Massachusetts Public Health Labs, we developed a lyophilized controlled release formulation with aluminum phosphate. So these are the like adsorption parameter, which I think Harm already went over these. I am not going to spend time on these. So these are again effect of anions on desorption, again from the work of Stanley Ham's group. And you can see that as the ion concentration increases, particularly phosphate, the percent of aluminum adsorbed start getting desorbed. Again, I think we already saw this slide again from Stanley Ham's group that the phosphorus content affect the point of zero charge. Actually, I take it a good point and make it versatile. You can make whatever you want depending upon the like charge on your antigens and also whether you want the adsorption or not adsorption. I think this played a good role in the diphtheria, adsorption of diphtheria, how much you want and whether you can manipulate it. Again, another some more data on the same thing. I think I'm going to skip that also. So this is some work we done at the Massachusetts Public Health on the effect of pH and phosphate ions on adsorption. So you can see that diphtheria toxoid is the more sensitive to these conditions. So PBS at pH 7.2, you cannot resolve any diphtheria onto that. But if you change the pH to 6 to 6.3, you get 57%. You change the PBS to saline, you get some adsorption at 7.2, but almost complete adsorption at 6.0. So if you use this was the in situ. So if you use the freshly prepared, I would say almost similar results, but a little better than the in situ. And Adjuvants showed almost similar characteristics, but a lower adsorption in the presence of PBS, and even at lower pH as compared to the gel made at the Massachusetts Public Health Labs. And aluminum hydroxide binds strongly because it has opposite charge to the diphtheria. So at any condition, it is almost completely adsorbed. And tetanus, usually there is no problem. So this is another experiment, effect of phosphate ions and the pH. So when you have a pH again with the, I think it is again with the diphtheria toxoid, because diphtheria toxoid is much sensitive to these conditions. So at 1x pH 600%, with the same condition, increase the pH, adsorption goes down, and increase the phosphate concentration, pH 6, adsorption goes down, and raise the pH, more phosphate, adsorption goes down, goes to 0, and with the 10x, bring the pH to 6, the adsorption, there is some adsorption. This is another slide from Professor Stan's lab, Callahan et al., and making the same point that on the malaria antigen, the effect of pH on the adsorption. So based on these studies and literature, I think this list suggested parameters for adsorption. Based on my experience, I would say that first of all, avoid ligand exchange by using aluminum phosphate or phosphate buffer with the aluminum hydroxide. So you should have an opposite charge on antigen and adjuvant at formulation pH, which is sometimes difficult to obtain, particularly formulating DTP vaccine on aluminum phosphate. That's why the DTP is formulated close to pH 6, because there you get some adsorption and the charge is not quite opposite, but you get some. So the other forces, hydrophobic, van der Waal, hydrogen bonding, play some role, but not enough for binding if there is same charge on antigen and adjuvant. And pH you optimize, again, usually it is done from 6 to 7.5, due to like you don't want to go too low than 6, otherwise the vaccine will sting on injection. And the size of gel particle, as I mentioned, should be preferably less than 10 micrometer, but it increases depending upon the what vaccines you are putting on. And ironing strength recommendation is low, absence of phosphate ions. So ligand exchange, I will not go into what it is. I think you already heard very elegant description from Harm. So my experience with the Hib conjugate, hepatitis B, and one other vaccine, which probably I would not name because this I learned as a consultant and maintaining confidentiality, but all three behaved very similarly. When you have this, the vaccine did not work. Low potency, immunogenicity, even in humans. I think for me, a quick test to see that your like formulation is going to work, you adsorb. If it is a ligand exchange, you will not be able to dissolve the adjuvant. The antigen will not be released. Whatever you do, you treat with concentrated sulfuric acid, bring it to pH of 2, it will not dissolve. So I think what it does that it may distort secondary and tertiary structure, changing epitopes. And antigens may not be accessible to antigen presenting cells because they are so bound tightly to the aluminum adjuvant, and they are not presented to B cells, and they are not processed by dendritic or other antigen processing cells. So how to avoid the ligand exchange? Convert aluminum hydroxide to aluminum hydroxyphosphate by treating with phosphate buffer. Phosphorylated antigens cannot replace phosphate actually, and you get stable and immunogenic formulation. I think my observation is that I don't know, some either unintentional formulations were done where it was claimed that it is aluminum hydroxide, or it was proprietary to keep the competitor confused. And in fact, it was aluminum hydroxyphosphate because the manufacturer was using phosphate buffer during formulation. So I think we all know that they have aluminum compounds give Th2 type adjuvant based on animal studies. They are strong adjuvants in animals. Based on my knowledge and experience, I think they are not good adjuvant with the humans and also with the other vaccines in animals, particularly influenza vaccine, polysaccharide protein conjugates. So I question the adjuvanticity of the aluminum adjuvants in humans, particularly based on data for tetanus and diphtheria vaccines, because during 1970s and 80s, there were a lot of reactogenicity to DTP vaccine due to whole cell pertussis vaccine and aluminum adjuvants. And in Sweden, they started using unadjuvanted DTP vaccines. And they did not see any effect of that in the increase of tetanus or diphtheria disease, or the immunity to tetanus diphtheria was almost similar. Then I think this data is again not published. The pneumoconjugate vaccine, I think it was either 9 valent or 11 valent. A small study was done in humans with and without adjuvants, and there was no difference. And recently, Merck's 21 valent pneumococcal vaccine has been licensed, which does not have aluminum adjuvant. And also, the role of degree of adsorption has been questioned in humans. So I think based on all these observations, I'm not sure that if aluminum adjuvants are actual adjuvants in humans. However, they provide good formulation vehicle for low quantity antigens, particularly if you are not doing any formulation work. So these are some of the articles from Sweden which show the IgE after injection of vaccine on aluminum adjuvants. So in animals, actually, they were great. Degree of adsorption is very important. So if you don't have any adsorption of diphtheria toxoid, no response. You have 90% adsorption, very high response. And then you go to increase the antigen, the low adsorption, then even lower, but you increase the adsorption and the adjuvant, the response is very high. And well, as I said, that aluminum hydroxide, there is always 100% adsorption, but the response is dependent upon the dose of the aluminum adjuvant. Particularly if you see zero point 0.1 adjuvant, so you have the highest response as compared to 0.033 at the same dose of diphtheria, 1.5, and these differences were statistically significant. Some comments I want to make that aluminum adjuvants have been used as benchmarks for new adjuvants because there has been search for new adjuvants for almost I would say 70, 80 years. I remember that Maurice Hillman was developing a adjuvant 65, which was based on the oil-based adjuvant and oil emulsion, but it showed great promise in animals. And then all of a sudden, the publication stopped. So when I met Dr. Hillman at one of the meetings, he said that it did not work in humans. So you know that negative data is not published in journals. So this was actually the breakthrough came when Gary Oat and Gary Van Nest and Derek O'Hagan developed MF59 adjuvant, which is now used in human vaccines. So I think my point is that these have been used when they are used as benchmark. They have been used as suboptimal formulations. Sometimes it is mentioned as alum. And in preclinical studies, as I mentioned, strong adjuvants and some of these animal models has limited utility, not reliable models for disease. And many times these immunogenicity experiments are not optimized. There is no dose response, very high dose is given. Sometimes with the model antigens like 100 microgram is given in animals. And sometimes the adjuvant is diluted, which I don't recommend, and I will show you some data on that. And mouse strain is important if you are going to study the IgG subclass. And a few comments on the selection of the methods, expressing the results in titer and all that. If you want to do a good study with the adjuvants, maybe you have to pay attention to some of these conditions. And these are described in this detail in this article. published in Vaccine in 1995. So this shows that if you dilute a vaccine and inject to animals, you don't get much response. Actually, this is like 1/50th and 1/100th of the human dose, which is on the dose response curve. You can see that with the 1/50th, you get IgG after one dose 3.3, at 1/100th 1.29. So, but these are when they were injected undiluted in microliters, 10 microliter, 5 microliter. But when they were diluted in saline, there was almost no response. So how do you inject those in animals? That also important. And I think Lauren talked about some of the side effects. This slide lists the side effects. I think there was much discussion about the side effects during 70s, 80s, and 90s due to the pertussis vaccine and aluminum adjuvants. But looking at the track record of vaccines containing aluminum adjuvants, I conclude that aluminum adjuvants seem innocuous for humans, and the aluminum is cleared through kidneys. So a few comments about the depot formation. So Glennie proposed depot formation at the site of injection, which was disputed by Holt in 1950 when he actually excised the depot from the site and the animals continued to produce antibodies. And we did some work actually not to study the aluminum adjuvants. We wanted to see the where the PLGA microspheres go after injection. So we prepared a C14 labeled tetanus toxoid by detoxifying the toxin with the C14 labeled formaldehyde. And the aluminum actually antigen on the aluminum phosphate disappeared within 24 hours versus 40 to 50% for PLGA and PLA. And also a study was done by Flarand and Ham with the aluminum 26 in rabbits. They also concluded that probably it should disappear from the site of injection quickly. And then I had some unusual observation which also dispute the depot formation. We were studying various adjuvants, and one of the adjuvant was sterile tyrosine, which bind the antigen very tight. So unfortunately, I think I will not go into details. We made a tetanus toxoid from purified toxin, and it reverted actually in vivo, which is a kind of scary. So when we injected into guinea pigs, the guinea pigs injected with the aluminum phosphate TT and fluid TT started dying on 10th day, but none with the sterile tyrosine, which was binding to the toxoid or reverted toxin tightly. So that also shows that the aluminum, the tetanus toxoid or toxin, the reverted toxin, does not stay at the site of injection. But there were some conflicting results from experiments by White in 1955 and 67. And you also heard some new data from arm which support the depot formation. So I would just leave it there that it's still not clear what is going on. So this is the a study with the radio level tetanus toxoid. You can see that the fluid TT and the microspheres, two different sizes, plus TT physical mixture, the radioactivity disappears quickly from the site of injection. With the aluminum phosphate, at 4 hours, there was good amount. So it stays there little more than the fluid toxoid, but then at 24 hours, it's almost completely gone. And with the microspheres, you can see that actually this is not a 24 hours, this is 24 hours. This was probably 12 hours. With the microspheres, you can see that it stays around 20 to 30% up to 4 weeks or 6 weeks. And one interesting observation was that aluminum phosphate crystals were still at the site of injection in most of the guinea pigs and mice after 1 year. So this work has been actually published in a vaccine. The article is listed in the list I showed you earlier. And this is the distribution of tetanus toxoid in lymph nodes and kidneys. I think kidney showed some at 4 hours with the aluminum phosphate and fluid TT, whereas with the microspheres, it peaked at around 7 days. Similarly, in lymph nodes, I think microspheres had a later at one day, but with the aluminum phosphate and fluid TT uh, was there for a few hours, then it disappeared at one day. So all the details of these articles, these studies are published in the journal Vaccine. Then a few comments on the aluminum compounds with other adjuvants. I know that Derek is going to talk about it. I'm not going to steal his thunder, but just that historically it has been used with the DTP with two potent adjuvants in whole cell vaccine, pertussis and LPS. And Carl Elbing in 1993 described it with the liposome and MPL. And then I think GSK is working on different formulations and the aluminum adjuvants. And Peter Cooper, actually, with whom we collaborated, had a gamma inulin mixed with the aluminum adjuvants. He called it algamulin as an adjuvant. Cancel described with QS21. And recently, Bharat Biotech actually had a new immunomodulator, mediajokinolin, with the aluminum adjuvant, and which adjourned to the aluminum hydroxide. So my thoughts on what's going on with the other adjuvants. Aluminum compounds bind tightly to other adjuvants through ligand exchange. I think Laurent already mentioned. Probably hydrophobic interaction and ionic also. Do aluminum compounds play a role as adjuvants? I think some study should be done with and without aluminum compounds if somebody really interested. But I have not seen any good clinical studies. I would be interested if anybody has a good clinical study showing that in humans, aluminum compounds are good adjuvants. I think the advantage of using other adjuvants with the aluminum adjuvants due to their binding, they reduce the side effects. I think Laurent mentioned that the LPS gets bound and you cannot detect it. Probably it will also reduce its pyrogenicity. And because these new adjuvants are very potent immunomodulator and have side effects. So that's the advantage of putting with the aluminum. And another observation is that sometimes they produce antibodies to the adjuvant in animals and humans, particularly monophosphorylipidae. Will not go into the problems, but this can be an issue. So finally, a few comments on the regulations on use of adjuvants. 21 CFR 610.15 constituent materials, ingredients, preservative, diluents, adjuvants says that an adjuvant shall not be introduced into a product unless there is a satisfactory evidence that it does not affect adversely the safety or potency of the product. So it is generally expected that an adjuvant should only be used with the vaccine if it is required for effectiveness and safety of the product or provides a clear or distinct benefit. The need or benefit of using an adjuvant with a vaccine must be demonstrated in humans, as animal studies have not been good predictive of effectiveness of most human vaccines, particularly vaccines used with adjuvants. Nevertheless, aluminum adjuvants have been approved with a number of vaccines by convention based on adjuvant TCT to animals, often without a clearly demonstrated enhanced immune response or protection in humans. As I mentioned that the recommended doses are from 0.85 to 1.25, but usually they are used at very low as compared to what is advised or what is required. And aluminum, I think aluminum adjuvant has been considered innocuous as FDA does not ask for need to include aluminum adjuvants. They take as it is. And adsorption, we also talked about that it should be more than 80% or more than 75%, but it is not enforced. And I know about it because one of the US manufacturer of DTP vaccine at a WHO workshop, which they were discussing to set this requirement, objected to that. They said that their diphtheria almost does not adsorb at all, but their vaccine has been effective for almost 50 years. So that was the point when the WHO dropped this. So this WHO requirement actually, it is not a requirement. It is a recommendation in a WHO manual, which is just a suggestion. But this is a requirement, but I don't think the FDA enforces it because I know based on the information provided by that manufacturer, their diphtheria toxoid does not adsorb up to 75%. So what are the regulatory expectations?
And Rajesh,
Build-
sorry, just a couple minutes to keep on
Yeah,
time.
I'm done. I'm done.
Okay, perfect.
Everybody was taking more time. I thought otherwise I would have stopped at 40 minutes if everybody stuck to their time. Building quality in the product is important. Quality by design principle should be followed. Validated manufacturing process is required, and well-defined and well-characterized aluminum adjuvant and formulation is required, and which should be the same as used in the pivotal efficacy trials. Consistency in manufacture is very important with the aluminum adjuvant as also for formulation, which should be demonstrated based on the test on aluminum adjuvants and adjourned product as I shown earlier. So aluminum adjuvants have almost a century of use in humans with a good safety record. So I don't have any concern actually, even based on my personal experience that when I took vaccines adjourned onto aluminum adjuvants, there was some local pain and redness or all that. But I had that more local reaction when I took the Shingrix vaccine and the COVID-19 vaccine. So from there on, I said, no, aluminum is much better. So it is no more a dirty little secret or art, thanks to Professor Ham. Physical and adsorption properties depend upon how these are made. Manufacturing and formulation affect characterization and adsorption. Different products at different manufacturers, probably not by choice. Maybe that's the way they have been making it. Strong adjuvants in mice and guinea pigs. Probably if we are making vaccines for mice and guinea pigs, probably these are great adjuvants. But unfortunately, we are making vaccines for humans. So I'm not sure. But they have a role as a formulation vehicle if you don't want to spend time on formulation of your vaccine. And it is a good base for other adjuvants actually. Adsorption again is important in mice and guinea pigs, not in humans. If they are not good adjuvants in humans, the adsorption does not matter. Benchmark to evaluate new adjuvants in animals. Depot information at the site of injection has been disputed. And the key message for regulatory consideration is consistency in manufacturer, particularly to those lots which were used in the pivotal clinical trials. Thank you.
Thank you, Rajesh. Questions, please add to the chat and we will go through them later. We have a 5 minutes break and we will start right after 5 minutes. Thank you so much. Anything to say, Laura? Anything to add?
Nope. Yeah, we'll come back in 5 minutes to reconvene.
Okay. Okay. Thank you.
break. Okay.
Are we ready, Laura? Another minute.
Lakshmi, can
Laura?
you hear me?
Yes, I can hear you. Thank you.
Thank you.
And
We can
you'll be
start
able to take
whenever.
control.
I think we can start whenever you're ready, Bob.
Ah, okay. Thank you.
Okay. Okay, our next speaker is Dr. Bob Citron, and he'll tell you all about aluminum adjuvants and his experiences. Go ahead, Bob.
Hi. Yes. Hi. Good morning, everyone. And thank you for attending this seminar. And thank you to the speakers this morning and to Lakshmi for organizing this. I've been working for many years with a variety of vaccines on aluminum, and it's always a little more complicated than one thinks. But over time, I've had developed a profound respect for the complexities of aluminum and aluminum vaccines. And I'm going to share with you a little different than the prior presenters, some experience I had when I was at Merck on a bunch of vaccines, which were on aluminum, and how we resolved or improved them as best we can. Going to talk today first about alum versus the AAHS adjuvant, which was mentioned before. Give some case studies in particular with the phosphate-containing antigens, the Hib vaccine and the hepatitis surface antigen. And separately, independently talk about how we optimized the adjuvant for Gardasil. Recall that the Hib vaccine and the hepatitis antigen vaccine were from times past, and the Gardasil was a more modern vaccine. And finally, end with some thoughts on how best to measure the potency of recombinant proteins absorbed to aluminum. And the first topic is alum. I'm not going to belabor this again, except to state that alum, of course, is not an adjuvant. It's a chemical. It's kind of slang for use, particularly by the clinicians. But in reality, of course, you've seen the discussion of aluminum chemistry several times. If you dissolve aluminum salts in water, they're very acidic. The pH is 3 or so. And when base is added, the pH rises, of course, and the aluminum comes out of solution into this precipitate. And again, aluminum hydroxide, you have a soluble salt such as aluminum chloride. You add a base and autoclave to sterilize, enhance the reaction time. And that was discussed at great length from the presentation from Croda. I will say that the making of aluminum adjuvants is a complicated process, and the best thing to do is to buy them and not make them in-house. Aluminum phosphate, of course, has the phosphate added to it, and I'm going to go into this a little more in detail later, but the precipitate is not a stoichiometric formula, and it's always a mixture and is also autoclaved to sterilize. What about alum? Alum is potassium aluminum sulfate. It's not an adjuvant, it's acidic, it's a very acidic salt. It cannot be injected. And historically, it was a convenient source of aluminum for the early preparation of adjuvanted vaccines, including work that was done at Merck. And we ascribed the name to the actual adjuvant that was prepared as amorphous aluminum hydroxyphosphate sulfate because of all the ingredients that were in there. It is amorphous, and I'll show you some EMs. You've seen them already. Obviously contains aluminum. It is a mixture of hydroxy and phosphate, and actually has about a small stoichiometric amount of sulfate in it, which makes it kind of unique over adjuvant or aluminum phosphate. Maurice Hilleman, who was worked on the very early vaccines, used that procedure for making Heptavax, which is the serum-derived hepatitis vaccine, Recombavax, the recombinant one, and later on we used it for our Hib vaccine, and I'll go into some details on those vaccines as we learn more about their interactions. Co-precipitation was the method used in the 80s, first described in 1948 by Cabot and Mayer. And in that particular case, described in that book chapter, you dissolve the antigen in PBS. And it's interesting, PBS, phosphate buffered saline, is commonly used. So it's the phosphate in there that was the magic ingredient in these. And then you add alum, the pH goes to 3, and then sodium hydroxide to neutralize it. And everything comes down into a supernatant, and the ratio is between 0.3 and 0.4. You can see both numbers quoted, versus around 1 for aluminum phosphate and adjuvants. Since the antigen is in there, it cannot be autoclaved, and therefore it is all done by aseptic processing. I would say, though, that this is co-precipitation is very hard to scale up and can be a problem. And I show this, the Croda presentation did not discuss the complexities of aluminum phosphate, but the binding of aluminum for phosphate is tighter than the binding of aluminum for hydroxide. What we have here is a use curve. The red line here is the addition of sodium hydroxide from basically 0 to 100%. And this is in solution what is going on. And you can see that the phosphate comes down faster than maybe the protein or the aluminum. And what this means is if it is not done in the same manner at different scales, you can imagine that there is a core of aluminum phosphate and a surface of aluminum hydroxide. This can make complexities because the later part of the precipitation is aluminum hydroxide and the first part is aluminum phosphate. Stoichiometrically, it's the right material, but as the scale changes, there could be problems. Nonetheless, this does work, and it did work very well for the vaccines in question. And this is the EM diagram. This is the EM diagram for EM for the aluminum hydroxide, and that's aluminum phosphate. And you can see here the Merck aluminum amorphous aluminum hydroxysulfate, and you can see that it basically is an aluminum phosphate. The slides, I'm getting a note that the slides are not getting updated. I'm seeing them are. Laura, is the presentation working?
Do you not have control anymore?
I do. It's working, but somebody reported they're not seeing. Are you seeing?
I can see it. Okay.
Okay.
It
I'm
looks
sorry.
okay.
I'm getting messages. All right. Thank you. Okay.
Working.
So
I think it's working on my computer.
okay. Thank you. Co-precipitation is very hard to control and scale up, and the actual material could change. And the modern method, of course, is to absorb to pre-made adjuvant. But these vaccines that were made in those days, that's what was commonly done. I only warn those of you who choose to make your adjuvant in-house. Any change in scale or equipment will affect the nature of it. And I strongly suggest leaving the manufacturing of adjuvant to the experts. I'm not getting a commission from Crota, but I'm just stating that, trust me, this is much more complicated than can be observed. So let's talk about some issues now that were observed, not with regard to scale up per se, but with the vaccines themselves on the aluminum. And of course, the one I'm going to talk about first, and it was mentioned already, is the hepatitis B surface antigen, which is either the natural or recombinant protein, where as it's expressed in the host cell, it attaches itself to phospholipids, which it pulls from the cell to make a spherical protein vesicle, a 20 nanometer particle. And all the commercial hepatitis B vaccines, except maybe some modern ones, are put on aluminum adjuvant. And the Merck HPV vaccines were developed by Maurice Hilleman in the 80s and were co-precipitated as per Cabot and Mayer, and they work quite fine. And the primary binding mechanism was ligand exchange. Recall, this is not adjuvant, so there are openings for more phosphate binding to the aluminum particle. As time went on, the world wanted to develop combination vaccines, which are now the norm, where instead of giving monovalent hepatitis B to children, it was included with DTP, Hib, and polio. And as you could imagine, there was immune interference, and there required to be more boost for the various ones. antigens in there, including hepatitis B, which worked quite well in monovalent, but was under stress in this mixture. So we did some work where we changed the phosphate levels in the AAHS and doubled it. And the antigen we found, and I'll show you the data, binds less tightly to the higher phosphate. And we could actually measure that by adding fluoride, which binds even more tightly to aluminum than the others. And one could measure the displacement. And we found that the higher phosphate adjuvant made the vaccine more immunogenic. And this is the data we observed. I pulled this from the paper. Samples of hepatitis B vaccine were prepared by co-precipitation, but with higher amounts of phosphate. And you can see the GMT, now this is in mice, went up significantly, and the percent binding, these numbers are real. The tightness of binding went down in the sense of that material was more readily displaced. What's really striking is the hexavalent, and these are two different hexavalent combination vaccines with and without the hepatitis B previously prepared with at the lower and the higher phosphate levels. And you can see striking differences in mice in the formulation A. Formulation B, you also see a difference, and it's interesting. Formulation B actually has extra phosphate in it for other reasons. And you can see the effect is less striking, but pre-treatment of the hepatitis B with the phosphate still indeed made it more enhanced. And this is the process used for these vaccines at this time. Another class of vaccines where we wanted to explore and talk about now is the issue of polysaccharide conjugate vaccines, in particular Hib vaccines, which contain PRP, but this also applies to some of the pneumoconjugate. Aluminum, as we've said, will bind tightly and catalyze depolymerization and loss of epitopes. This has always been an issue for PRP Hib vaccines. Originally, they were administered as unconjugated, and then some of the early conjugate vaccines were found to be unstable. When the early work was done, in order to work around this, these vaccines were lyophilized to be stable. You've seen this chart before. Phosphate on an antigen reacts with an aluminum hydroxide entity and displaces hydroxide, and this bond is more tightly observed. This is the polymer of PRP. And what is really interesting, this ribosyl phosphate is kind of like RNA. It's got this hydroxyl group near the phosphate, and that's kind of critical. This is what PRP looks like. It's a polymer of ribitol ribosyl phosphate repeated in a molecular weight of about 100,000 or so. What we found in the vaccine we were working on is called PDVax-Hib. It's a conjugate vaccine of PRP and the outer membrane protein complex, a lipid protein vesicle. It's a spherical vesicle. The original product was lyophilized and gave excellent clinical data when it was reconstituted with our adjuvant. But in order to make combinations, we had to put it in surface and absorb it. And we noted that this material actually depolymerized over time. And that's what you can see here. What was going on is this hydroxyl group would displace the phosphate, which would go on catalyzed by the aluminum, and it would break the polymer. And this is the reason that PRP is actually unstable on aluminum surface. What was really interesting, and this is pulled from the paper, whereas it was fully conjugated, the 100,000 molecular weight PRP was in the conjugate vaccine. Over time, it dropped down to about 80% and then became stable. it didn't completely depolymerize. We discovered this, and luckily we had clinical data over this entire range and didn't see a major issue, but we were very perplexed with how it could start decomposing, that is depolymerizing, but not go further. And it was stable after that first drop for as long as 2 years. This was not considered an issue for the clinic because the clinical studies span early and late time periods. But we do know that PRP is labile, as I mentioned. So why did it stop at 20%? We imagine that the vaccine is a sphere and could only contact the part of the aluminum on maybe 20% of its surface, and maybe that's where the aluminum caused the deconjugation. The rest of it was protected from the aluminum. I think in general, in the future, for certainly vaccines like this, aluminum phosphate would be a much better antigen, a much better adjuvant for these type of phosphate containing antigens than aluminum hydroxide. And in combination vaccines, this has actually been shown and claimed in the literature. And aluminum phosphate is the preferred adjuvant over aluminum hydroxide for pneumoconjugate vaccines, presumably for these reasons. And 19F, for example, could be labile to the same deconjugation. Okay, completely changing the subject. Now we're going to a modern vaccine. And how does one optimize the adjuvant for a vaccine, Gardasil? This is work that was done with the original licensure. It gave us the opportunity to explore, and clearly there was some mention earlier in other presentations of why you really need aluminum. And the FDA, of course, insisted that we provide data justifying it. And we did our work in primates and could show, and this was published, that the antigen response was increased to tenfold using aluminum adjuvant. The question was which one? We could use aluminum hydroxide, aluminum phosphate, or our own. And I point out that we also knew that the highest stability for the HPV VLP antigen, and HPV is a particle, about a 60 nanometer particle, which is most stable at pH 6. And in this particular case, we were finished with co-precipitation. We used preformed adjuvant, which was made, as we showed you before, but just with no protein in the precipitation. This is a surface charge profile. This slide was presented before. My only point is the amorphous aluminum hydroxyphosphate sulfate is its point of zero charge is near neutrality, meaning that the surface pH is close to neutrality, whereas the others have, as mentioned before, more extreme surface pHs. And so there is a possible concern that the antigen would be in an environment it did not like. We also studied the surface capacity binding, and you can see the AAHS also binds more material than the other two. Of course, we had to have a safe binding capacity for our antigen. But this was the most telling study, the work we did in BELPC mice. And this is a log scale on the left, and you can see that clearly the AAHS and adjuvant was more immunostimulating, I guess, although close to aluminum phosphate and clearly better than aluminum hydroxide. And so my suggestion is anyone who is still developing HPV vaccines, and there are still some, aluminum phosphate is clearly a better adjuvant than aluminum hydroxide. And of course, these three adjuvants differ in chemical properties, and we believe that part of the reason is stability, which we have seen where the VLP is most stable at closer to pH 6. where the surface pH for the AAHS is better. Certainly, even the aluminum phosphate would be somewhat better as well. And as I stated, we use that pre-made. This is not really important to the discussion, but it's pretty. I like to see pictures. This is really compelling. You can see in here a cryo-EM that we got of Gardasil on the adjuvant, and you can see the amorphous plates of aluminum adjuvant here and the beautiful HPV particle here. And you can see the sitting on the aluminum surface. We find these pictures very useful and telling us, showing us how intact the VLP particle is on the aluminum surface. I want to close with one other item. In the past, animal potency has been used to measure the immunogenicity of protein vaccines, and the world is now moving more to in vitro potency. There is an issue with in vitro potency, although that was used for these vaccines for hepatitis antigen and the HPV vaccines at their time of either for HPV at the time of licensure and for hepatitis antigen over time, the animal test has been eliminated for in vitro. And the way you do an in vitro potency test is the antigen is desorbed with a buffer, usually phosphate or citrate, which dissolves the aluminum. And the freed antigen is measured by a sandwich ELISA. And this is all expressed in a relative potency format against the standard. This was a breakthrough, certainly away from animal potency, and gave tighter data. And this is the general norm now for hepatitis antigen. And as I told you, when Gardasil was licensed, it was actually licensed with the in vitro test. There were some issues that were observed. And it was mentioned earlier by Harm's talk that over time, these proteins tend to nestle into the aluminum and get a little harder to desorb. So for older samples where binding gets tighter over time, the desorption can be more difficult and may require more aggressive treatment. If you look at without completely desorbing the antigen, this could result in an underestimation of the potency because less is recovered, or in a rather sinister manner, overestimated if less is removed from the standard. When we were working on this years ago, my clinical people argued that my potency assay was not good because, unlike an in vivo potency assay, the potency test is not done on the vaccine itself, but on a desorbed antigen. We published extensive work to show that the in vitro test did indeed correlate very well with animal and human clinical data. However, in more modern vaccines, we've had a chance to revisit this. And we have developed here at PATH a different method of measuring doing the ELISA on aluminum vaccines, which I want to share with you because I strongly recommend this to any of you who are working on a vaccine on aluminum. The vaccine, while still on aluminum, is pre-treated with a blocking agent to minimize any non-specific absorption. And then the absorbed vaccine is diluted on a plate in the presence of a constant amount of a monoclonal reagent that is specific for the antigen. The antibody binds to the antigen on the aluminum surface. You recall one of the other speakers talked about changes that occur for the protein on the surface. It could degrade or otherwise undergo degradation, oxidation, deamidation, etc. And the antibody, which should be to a protective epitope, could see any effect of that. So the the absorbed vaccine is spun down, and the supernatant, which contains the leftover, the non-bound antibody, is measured on an ELISA plate, a different plate, similar to the way you would do serology in a serology format using the antigen as a capture. The good advantage here is it only requires a single monoclonal. One does not need two antibodies for this. And the potency can be calculated by subtraction. You still run this in a relative assay format. And it actually works for combination vaccines if you have several different antigens, because the individual monoclonals will bind to only their paired material. This is an example of the work we did at PATH on a recombinant protein, which was a trivalent, non-replicating rotavirus vaccine. This is on aluminum. And this particular vaccine
Bob,
is
you
a
have
mix.
about 3 minutes, sorry.
Yep, this is it. Thank you.
Perfect.
This particular vaccine is absorbed onto aluminum and is a mixture of all three. And in this slide, you can see that when we ran this vaccine in this particular case, we were testing for the P4 antigen. You can see either the free material, the P4 on aluminum, or the P4 in the presence of the others did give this curve. Notice that as you dilute it out more, the signal gets larger because you're doing a subtraction. We were able to validate this assay and in fact used it by some of our partners for a COVID vaccine in India. So I would like to acknowledge again Stanley Hem and his lifelong work on understanding aluminum adjuvants. I do miss him. My colleagues at Merck, where we did most of this work, and Jessica White at PATH, who developed the inhibition ELISA. And thank you for your attention.
Thanks, Bob. That was awesome. We'll move to the next talk by Christopher Fox on the formulation strategies to enhance immune response quality to aluminum-containing adjuvants. Chris?
Thank you. Yeah, thank you, Lakshmi, and to the other speakers. It's a pleasure to be here. Really enjoyed the talks so far. I am a formulation scientist, and so I'm going to talk a little bit about the formulation aspects of aluminum-based adjuvants. And let's see. Thank you, Laura. So as you've heard today, aluminum salts have been used for many decades in vaccines for humans, and they have an excellent safety record. Some of the practical advantages of aluminum-based adjuvants is that they facilitate single vial liquid formulations of antigens absorbed onto the aluminum salts and potentially other adjuvant components, making administration straightforward for the end users. As you've also heard today, the particle size of aluminum salts consist of heterogeneous aggregates ranging from nanometers to several microns in size. Unfortunately, this particle structure is not stable to freezing excursions. And so it's been documented that many vaccines lose potency if they contain aluminum and have been frozen. So this is a liability of aluminum-containing vaccines. Again, as we heard earlier, in some cases, aluminum salts may not provide a potent enough adjuvant response, depending on the indication and vaccine antigen of interest. And I'm showing here an image of the issue with freezing. On the left, you have a frozen aluminum-containing vaccine vial, where you can see the larger particle structures both in the EM image and in the sedimentation of the vial. So the good thing about this is that it's obvious to see when an aluminum vaccine has been frozen. and it can therefore be discarded afterwards. Sorry, can we go back one slide? Back one, please, Laura, if you can. Sorry. I think we're probably hitting the button at the same time.
No, I also can't go back. Sorry. There we go.
Thank you. So to address this question of the cases where the adjuvant activity of aluminum is too modest, let's say, where we're looking to increase either the quality or the magnitude of the response. One approach is to add additional adjuvant components, such as TLR agonists. And so there are several cases in the literature involving clinical data and many more with preclinical data indicating that adding a TLR agonist, usually adsorbed to the aluminum salt, can potentiate the quality or magnitude of the immune response. And of course, this depends on the animal model and the antigen and the formulation. But some examples here regarding clinical data on the left with a recombinant malaria antigen, where the antigen was adsorbed to aluminum hydroxide with or without the TLR9 agonist, CpG 7909. And you can see that after 2 immunizations on day 42 or after 3 immunizations, the addition of the TLR9 agonist really increases the antigen-specific antibody titers. And the same was true for an HPV vaccine antigen developed by GSK. So now looking at the middle panel, where adding an adsorbed TLR4 agonist in the form of MPL increased antigen-specific antibody titers compared to alum alone. And so that's the comparison of ASO4 to the aluminum oxyhydroxide alone. And that was true as well in terms of B memory cells specific to the antigen, where you can see the increased number induced when MPL was included along with the alum in the vaccine compared to alum alone. Now, regarding T cell responses, you can find a lot of preclinical data indicating a very distinct shift from Th2 type responses, which are typical of aluminum salts in animal models, to Th1 type responses, depending on the TLR agonist that's added. The case is not as clear-cut in clinical data, and I'm showing here a very interesting study looking at many different adjuvants, actually. So now on the bottom panel, but I'm focused in here again on that same comparison between MPL on alum, which is ASO4, versus alum alone. And this is with a hepatitis B antigen. And really, you can see that the T cell response quality was quite similar. Really, there was no difference either in the quality of the T cell response or the magnitude in this study. They looked quite similar. So in humans, I would say the case is less clear-cut about that Th1, Th2 paradigm that we can more distinctly see in animal models. Okay, let's see. I'm having trouble advancing now, Laura. Apologies. Maybe if you could. Thank you.
So if you see the take control button, you can probably do it again, but if not, I can do it for you.
Yep. I'll take it back. Thanks. So how does one formulate TLR agonists with aluminum salts in an optimal manner? So much like you've heard with antigens adsorbing to aluminum salts, there are many considerations to take into account in adsorbing TLR agonists to aluminum salts. And the same mechanisms are at play here: electrostatic association or ligand exchange. And in some cases, it's fairly straightforward. For instance, in the case of TLR4 agonists like MPL, they already contain a phosphate group. And so they are inherently able to adsorb to aluminum oxyhydroxide, for instance, by ligand exchange. In other cases, the TLR agonist may not inherently have a phosphate or other functional group that adsorbs to the aluminum. And one innovative way around this is to modify the TLR agonist to contain a phosphate or phosphonate group, as was done by Derek's group at Novartis with the SMIP adjuvants, or small molecule immunopotentiators, which is shown here. They were chemically synthesized to contain a phosphate group, which would then adsorb to the aluminum. In other cases, which specifically one example, which I'll show you in a future slide here, you can use formulation approaches to, for instance, complex the TLR agonist with a phospholipid or other excipient that binds to aluminum and brings the TLR agonist along for the ride, even though the TLR agonist itself doesn't have that functional group to adsorb to the aluminum. Now, again, there are many considerations to take into account here, and it's important to characterize what is happening in this adsorption interaction and how it impacts the absorption of the antigen. So there are cases where if you adsorb a TLR agonist after the antigen has been adsorbed, it could possibly kick off the antigen. There might not be enough surface area for both components, so you really want to characterize that. And one group that's looked at this in an interesting manner using cryo-EM is Andrew Ward's group at Scripps, and they've indeed shown that depending on the adjuvant content and order of mixing, you can get undesirable effects on the antigen conformation. And so this is important to understand prior to going into your in vivo studies. Okay, so just a couple of examples in our work at the Access to Advanced Health Institute. We have a synthetic TLR4 agonist that we call GLA. The structure is shown in the top right there. And as with MPL, it has a phosphate group, and so it lends itself to adsorb to aluminum oxyhydroxide. However, it's insoluble by itself in water. And so the GLA needs to be complexed with a phospholipid to create a nanoparticle lipid suspension. And then that's what's mixed with the aluminum oxyhydroxide. And that lipid nanosuspension, those particles adsorb to the aluminum. But it's not just the GLA that adsorbs through its phosphate group, but the phospholipid also adsorbs because it has a phosphate group, of course. And so we showed that both those components adsorb to the aluminum. And in fact, on the lower left panel there, you can, if you look very closely, we think we can actually see those lipid vesicle structures adsorbed or hanging off of that aluminum oxyhydroxide semi-crystalline surface there, indicated by the white arrow. On the lower right panel is some clinical data, just reaffirming that adsorbing that TLR4 agonist to the alum gives you a stronger antibody response in humans. And in this case, we're looking at a phase 1 clinical trial of a recombinant schistosomiasis antigen adsorbed onto alum in blue, or onto alum with the TLR4 agonist in red. And only the TLR4 agonist group had significantly higher titers than the placebo in this particular readout. So another example that I'll go into a little bit more detail on here is with a TLR7/8 agonist developed by the 3M company. This is called 3M052, and its structure is shown on the left there, upper left. And you can see it does not have a phosphate group or other characteristic that would lend itself to absorbing to an aluminum oxyhydroxide. And so the way we went about this is by, again, creating a lipid nanosuspension, so taking an insoluble TLR agonist, complexing it with a phospholipid, and creating these lipid nanoparticles. But in this case, it's because of the lipid excipient that we're able to induce absorption onto the aluminum salt. And this really has resulted in some nice preclinical and now clinical data indicating quite a potent combination of 3-MO52 on alum. And I'm showing non-human primate data there in the lower left panel in terms of antibody secreting long-lived plasma cells, where you see a significant increase when you add the 3-MO52 compared to the antigen on alum alone, and that's with an HIV envelope antigen. To go into a little more detail on how we developed this formulation approach of absorbing the TLR agonist to the aluminum adjuvant, we initially started by, leaving the aluminum out of the question for now, and just how do we make a stable lipid nanosuspension with this insoluble TLR agonist? And so we looked at a number of phospholipid-based compounds as well as other common non-ionic surfactant excipients, and we complexed them with the 3-MO52 using standard thin film approaches with water bath sonication to create the nanoparticles. And then we measured the size of these lipid nanoparticles and the zeta potential. And really what we were looking for is a size that was smaller than 200 nanometers so that it could be sterile filtered after production. And so in the upper right panel there, we're able to really hone in on the lipid excipients that gave us the smallest particle size. And then we monitored that size over time to see if they were physically stable. And that enabled us to zero in even more on the two most stable excipients here, which one is DSPG, a phospholipid, the other is cationic lipid, DS-TAP. And sure enough, the zeta potential indicates that they provide the expected charge character to the nanoparticles. And this was interesting to us because now we had two lipid nanoparticle formulations that were electrostatically quite different. And we were most interested in the anionic composition because we were looking for a formulation that would absorb to aluminum oxyhydroxide in this case. So we selected DSPG as the excipient of choice here. And then we characterized the absorption of the lipid nanoparticles to the aluminum oxyhydroxide, in this case, alhydrogel from Croda. And what we found was that we see a nice absorption isotherm, and because the doses of the adjuvant are quite low in preclinical and clinical settings, we're confident that essentially 100% of the 3-MO52 is absorbed to the antigen, to the aluminum, and there's plenty of room left over for absorption of antigen on the aluminum salt. So it becomes a nice formulation to pair with different doses of antigen as the requirements might dictate depending on the indication. And when we increase the amount of salt in the formulation, we do not see the absorption levels go down. So we think this is likely a ligand exchange mechanism where the DSPG, the phospholipid, is binding to the aluminum, and it's really bringing the 3-MO52 with it based on hydrophobic association. And here's some preclinical data where we really looked carefully at different iterations of this. Okay, so is the combination really giving you something more than either component alone, whether it's the TLR agonist alone or the aluminum salt alone, and to look at whether the absorption was important for activity, we compared it to the same TLR agonist mixed with aluminum phosphate, which it does not bind to. And so those are all the groups in this study. We used a recombinant HIV antigen again, and we looked at both antibody and T cell readouts. So on the lower left here, the serum antibody titers, you can see we're highest with the orange bar. That's the 3-MO52 on the L-hydrogel, where it's fully absorbed along with the antigen. You can compare that to the 3-MO52 without alum in blue, or the alum without 3-MO52, the clear bars, or the 3-MO52 mixed with aluminum phosphate in green, where it's not absorbed. Now, of course, the antigen's not absorbed there either, but you see that absorption does play a role here. Also looking at long-lived antibody secreting cells in the middle panel, a similar story there. And then in terms of T cells, maybe an even more dramatic effect where we saw a significantly higher number of interferon gamma positive CD4 T cells with the absorbed hydrogel 3-MO52 formulation compared to the controls. Okay, so how about human data? So that's forthcoming. I'm showing you some hot off the press data that has just been generated and is in press. There's a preprint available online right now. And that is the part A of this phase 1 clinical study where because it was the first in human vaccine study with this formulation, we looked at 2 different doses of the 3-MO52 using the same dose of aluminum oxyhydroxide or L-hydrogel with a recombinant HIV trimer antigen. And what I'm showing here in blue is the low dose of the 3-MO52 at 1 micrograms, and then in red, the 5 microgram dose. And you're looking at data from individuals in this part A, so a small part of this clinical study. And as you might expect, we see antigen-specific antibody titers in the serum, actually both dose levels, and those increase with the number of immunizations. But in the right panel here, quite striking data where we're seeing that several of the subjects in the high dose group are demonstrating autologous tier 2 neutralizing antibodies, which are not trivial to achieve for those of you that are HIV vaccine experts. And so the study is now in part B, and all of the immunizations have been completed, and really it's the immunology analysis that's ongoing right now. And this is highly interesting in terms of adjuvants, because we're looking not just at this 3-MO52 L-hydrogel formulation, but we're comparing it to L-hydrogel alone, a TLR9 agonist on L-hydrogel, or a TLR4 agonist with a QS21 liposome, so without aluminum formulation. Along with the clinical data, which is forthcoming on this adjuvant comparison, there is a package of preclinical data in multiple different animal models, and we hope this together will be informative for not just adjuvant comparisons in general in a clinical study, but translating from animal models to the human setting, and what lessons can be learned there in general for adjuvants going forward. So stay tuned for that in the near future. So I've talked at length about adding TLR agonists to aluminum salts, and you'll hear more on that important topic on the next talk from Derek O'Hagan. There's another way that we've been working on to modulate the potency of aluminum-based adjuvants, and we call this nanoalum. And in this case, and there are other groups, several other groups working on this type of approach, we found that by modulating the physical characteristics of the aluminum salts through different processing techniques and or addition of excipients, We can generate different particle sizes, potentially different shapes of nanoparticles. And these have a quite striking effect on the immune response profile, but also the stability of the formulation. So specifically, in our nanoalum formulation, we're starting with alhydrogel, and we are breaking down the particle size through high pressure homogenization. And then in order to stabilize that smaller particle size, we do this homogenization in the presence of an excipient, in this case, polyacrylic acid, although other excipients can potentially be used. And through this excipient and process modification, basically what we end up with is these nanorods shown in the cryo-EM image, which, basically look like a broken up alhydrogel particle into much smaller nanoparticles. And these are physically stable over time. They do not re-aggregate. And perhaps most importantly in this respect is that you can freeze this nanoalum formulation and the particles retain their physical characteristics. So this is a very different from the traditional aluminum situation. When exposed to freezing temperatures. Even after multiple cycles, we see that the particle size is retained in the nanoalum. Now, in terms of immune responses, this is preclinical data in a mouse model where we showed that using a recombinant TB antigen paired with this nanoalum compared to traditional alum or alhydrogel, we found an increase in a Th1 type T cell response, indicated here by interferon gamma and TNF positive CD4 T cells. And we've seen this with other antigens now as well in different preclinical models. We're also working on potentiating this by combining TLR agonists with the nanoalum as well. So that's work to be done still. But we find that we definitely get a different response quality and magnitude by modifying the physical characteristics of the aluminum salt. Now this has yet to be tested in humans, so we'll see what translates there. But for now, we think it's a very interesting way to modulate the potency of the adjuvant activity of aluminum. And so summarizing, aluminum salts are, by far have been the most widely used adjuvant. And we're approaching the centenary mark now of when they were really first shown to have adjuvant activity by Alexander Glennie in 1926. They have an excellent safety record and are well used in many different vaccines, but they do have some liabilities in terms of the physical stability upon temperature excursions, and maybe not being potent enough for all indications. So we think there are ways to address that by pairing with TLR agonists, as I've discussed, although each antigen and indication has to be addressed on a case-by-case basis. This is not something that can just rely on sort of a blanket statement that it's always better to do it this way. And especially in humans, it's a more nuanced evaluation than what we see in animal models. In addition, some next generation approaches have to do with trying to assert more control over the nanoparticle characteristics of aluminum salts and really tune those for optimal stability and adjuvant activity. And while I think these are exciting, to my knowledge, they have yet to be tested in humans. And so that really needs to be looked at before we get too excited about it. Now, with the COVID-19 pandemic, I think, it was quite interesting for these types of approaches in terms of pairing TLR agonists with aluminum salts. And several new vaccines were authorized for use in humans and produced commercially, either containing TLR9 agonists adsorbed to aluminum or TLR7/8 agonists. And so I think that approach proved to be a great boon for development of vaccines that were potent and stable during the pandemic and widely used. But I think we have more room for improvement. So I'm excited where the nanoalum and other TLR agonist and maybe non-TLR agonist combinations take us in the future with aluminum salt. So I think the outlook looks very good, and looking forward to what we can do in future with aluminum-based vaccines. So I'll stop there. Thank you very much.
Thanks, Chris. I think you brought a lot of good points about thinking about the future of adjuvants and especially combinations to be able to get a better responses, especially the cell-based responses. I think with that we'll lead to Dr. Derek O'Hagan, who will provide us more insight into this topic. Derek, it's all yours.
I'm here, just need my slides to come up, and then hopefully I can get control of them too. So I believe I have control. Yeah. Yep, I'm good. So I'll, but I don't have